sir. If anybody is not there, you close the session.
Yes, Dr. Bag.
Yes. If anybody is not there, you close the session.
The importance of this assay lies in the fact that if you collect 100 samples, the probability of getting an EPN in one of them is like 4 to 10%. Okay, so 90% of the effort in the EPN is getting wasted. With this assay, you can efficiently recognize those 5 or 10 samples out of 100, and then you can insect your soil samples and isolate your EPNs from that type of product. So this is the utility of this assay. This has also been published by the board. Lastly, as Marao we have sharing this data for last 20 years. We have completed the draft of Melatonin glandular rice contributor, and this is final assembly.
Dr. Bag.
Yes, sir.
Dr. Prasad.
And
Dr. Prasad.
Yes, sir.
Is it what should you do? Only 5 out of 20, only 5 presented.
Yes, it is too less number, but anyhow, we can do anything.
So see, notification is up to 2 p.m.
Yeah, that's problem, na. Till 2 p.m. we need to wait.
We cannot close the session.
Then at 2 p.m.
Yes.
you have to start the next session 3.
Uh,
It has
no,
also scheduled time.
No, sir. Sir,
Time.
No.
You can start your session at the right time, but we cannot close before time.
Yes.
Because anybody can
Yeah,
claim
yes,
to
yes,
you,
yeah.
not to us.
Yeah, I came there, but it was closed. Why? Let me claim. But then one thing, we will wait for the just as such, we keep it mute and the video off. Then let's wait for up to 1:15.
140.
1:15. Then again check if anybody is there. Fine. Then we will start. Otherwise, again 1:30. After 15, every 15 minutes we will check.
I think in chat box we should leave a message.
That is.
We are waiting for all the participants till.
We are waiting for the participant session 2A and 2B. It is on. Session is on. Participants are requested to attend immediately.
Yes.
Like that. Can you share it with chat box?
Yes, we can write in the share box.
You write it.
Dr. Bag. Dr. Tushar.
Mr. Bag.
Okay, I can write, no problem.
Yeah.
I think we need to wait up to 1:15, then we can close.
Yeah. Let's see. Let's see.
similar position. There are third post regulation because this is the post splitting asset and this is the cooperative commercial injection asset. So in the post splitting asset, third
Anybody join here? I think slowly those who participated, they leave.
No, those are Prabha and some others are here. No problem. I think Dr. Saha,
Yeah,
in the technical session posters, there need to prepare a simple report and send to convener
yes.
as in open discussion.
Yeah, in the valedictory or whatever, maybe.
Madhubala Thackeray now joined
Madhubala Thackeray,
here.
Yes, sir. Yes, sir.
where?
So Madhubala, you present now to need to present.
Sir, please give me a few minutes to share screen.
Yeah.
Okay.
Okay, wait.
Sir, I hope we should wait till 2.
Yeah, it will be closed by 2.
Anybody join here? Press.
Yes, sir.
Madhubala Thackeray, 2B, session 2B.
Madhubala Thackeray join here, I think.
Yeah,
Dr. Saha and Dr. Prasad.
Yes, sir.
Hello.
Yes, sir. बोलिए.
Yeah. She is going to present.
Madhubala going to share their screen.
Lakshman,
Yes, sir.
are you getting my sound? Lakshman?
Yes, I'm getting your voice.
Yeah.
Ankita Sarkar also there now.
Yeah, fine.
Hello, Ankita. How are you late? Unmute, Ankita, unmute yourself.
Ankita Sarkar, you unmute it.
Yes, sir. Yes, sir. A very good afternoon to all of you, sir.
We are not getting your sound at all.
Yeah. Now I'm getting. Now I'm getting. Ankita.
Yes, sir. Very good afternoon, sir.
Very good afternoon. Very good afternoon. Are you getting me?
Yes, sir.
Yeah.
No sound is here from any side.
No, sound is coming now.
Then first Madhubala will present. Then Ankita.
डॉक्टर साहब फिर तो ये सेशन एक्सटेंड करना पड़ेगा.
Is it visible?
Yeah, you can expand it. Are you presenting through your mobile?
Yes, sir.
Okay, okay.
Is it visible, sir, now?
Yes, visible.
And voice is audible, sir?
Yeah, you are audible.
So, should I start?
Yeah, you started. Start.
Good afternoon. I'm Dr. Madhubala Thakre. I will present my poster on the topic study on different harvesting dates of Kinno under Delhi conditions. So, as we know that Kinno is a fruit which is very famous under North Indian condition because it is the only mandarin which can be successfully grown under North Indian condition. But the issue is that it is not, we cannot ripe after the harvest because it is a non-climactic fruit and it is not followed in this case. The rise in ethylene production after the rise in respiration rate is not present. It means it is a non-climactic fruit and it also indicates that it should be harvested at correct stage because if it is not harvested at correct stage, so after harvesting, it will not ripe and it will not attain its proper fruit quality. So for this purpose, the harvesting at right stage is very important. And keeping this in mind, one experiment has been designed with 4 treatments. D1 is last week of December. These treatments are with respect to harvesting date. So first, D1 is when harvesting was done in last week of December, D2 first week of January, D3 second week of January, D4 third week of January, with 5 applications and various fruit parameters, physical fruit parameters like fruit weight, diameter, length, and biochemical parameters like TSS acidity, TSS acid ratio, ascorbic acid content, total carotenoid content in juice and in peel was measured, as well as yield kg per tree. So when we see the data, it is very clear from the data is that under the treatment D3, it was found maximum TSS acid ratio, that is 14.733. Here, I want to inform that TSS acid ratio is very important with respect to non-climactic fruit, and for deciding the maturity is TSS acid ratio. So it should be more than 12. So here we got 14.733 TSS acid ratio with total carotenoid in juice was also maximum, that is 722.83 microgram per 100 ml of juice. So on the basis of these results, when we compare it with others, the D3, that is the second week of January, was found suitable for harvesting of Kinno fruit under Delhi condition. And as I already mentioned that second week of January was found better for Kinno harvesting in terms of higher TSS acid ratio, total carotenoid content in fruit under Delhi condition. And it means harvesting date for a particular locality with respect to Kinno is important, as it will help the growers to harvest during that period because they cannot measure the TSS or acid and cannot calculate the TSS acid ratio. They only harvest with the help of their visual or sensory observations. So this finding will help the kidney grower which are surrounding Delhi area. Thank you so much.
Yeah. Thank you, Madhubala. The very precise information. My question is, do you think the harvesting date, that is the best time is second week of January? Is it applicable in Punjab situation?
No,
Not necessary. Or Andhra Pradesh? No.
no, sir, it will not be applicable for Punjab or Ganganagar or other kidney growing area. It will be
Yeah.
specific. to Delhi.
Specifically, if it is specific, that's because this.
Sir, your
fruit
voice
maturity
is very low, sir. It is not
Yeah.
clearly audible.
Yeah. Maturity depends on the fruit setting time, flowering time, fruit setting time, right?
No,
Are you
sir,
getting?
it is, no, sir, I would like to say that it is Kinno sets fruit in the month of March, second week of March.
Yeah.
So it will be on the tree up to 9 to 10 months. So
Yes.
the peculiar period is winter where it matures and ripes.
Yes. That means 9 to 10 weeks it takes to mature.
No, months, sir, months.
Months.
You can calculate
Yeah.
from March, April, May, June, July, August, September, October, November, and then December, 9 to 10 months.
Yeah.
December and January. Very long period, sir.
Is there anybody who is working on this in Punjab or Andhra Pradesh?
Yes, sir. They are Andhra Pradesh, I don't know, sir, but in Punjab, people are working. In Hisar also.
Yeah. Okay. Okay. Any questions, sir?
Madhubala,
Anybody?
Yes, sir.
so as you are working with Dr. Kanhaiya Singh, M.K. Verma, Dr. Avasti, so
Yes, sir.
what is the problem with Kinno at IRI? What are disease or
What
any
is
other problem related to the pathology?
related to pathology? Sir, disease as such, disease is not a problem, but of course, phytophthora is there, and problem of decline is also there.
Decline,
That
Citrus
is
decline.
that is the fruit.
citrus decline. No fruit disease is not there.
Yeah. Post-harvest
Pardon,
disease.
sir?
There are any fruit post-harvest disease is there or not?
The post-harvest diseases means we are not working on that. We are working pre-harvest aspects.
No, pre-harvest aspects is okay. After harvest, fruits are fresh and healthy.
Yes, sir. Hello.
Okay.
Hello.
Then. Okay, Madhubala.
Thank you, sir.
Okay, sir. Okay. Okay.
Can I share?
Yeah, now you can answer your question. Now, another.
Ankita Sarkar is there.
Ankita Sarkar.
Yes, Ankita Sarkar and Afroz also. Ankita Sarkar, you start your share screen.
Is it visible, sir?
Yes. It's visible.
A very good afternoon to all the respected members, to all the delegates. Today I'm here to give a general presentation on the microbes and their role in post-harvest disease management of fruits and vegetables. Now we all know that the huge amount of losses are there in the field of fruits and vegetables due to post-harvest losses. Different data have also been presented, and different approaches are also being taken up for the management of the post-harvest losses, starting from the chemical and physical treatments, such as your high temperature, low temperature treatments, UV radiation, gamma radiation, chemicals are used. And along with that, now due to increasing demand for the use of green products, people are looking for the use of microbes in the management of post-harvest losses. Now, as we know that in case of diseases, there's a disease triangle which comes into play. Similar is the role in a case of post-harvest disease management.
Nikita, you can go in slide mode. Click
Sir,
on
no, sir, I have only a poster presentation, sir. So it's only in.
No. I mean that if you click on slide mode, then it will
Full.
be visible more.
Yes, yes.
Now
Yes.
it's okay, sir.
Yeah.
Yeah, you can increase the size just to maybe percent increase.
Okay, sir. Is it okay, sir?
Yeah, it's okay now.
So in this case, first, what are the causes? Several papers have been presented. I will not go into details of that. Careless handling along with microbial spoilage are the main factors which contribute to the losses. And talking about the aspect of biocontrol agents, that the biocontrol agent interacts with the microbe which is responsible for causing the spoilage and the host along with the environment for bringing about an interactive triangle. Here we must understand that the biocontrol agent is not the only factor which is playing, but it is interacting with the host and the phytopathogen in concern, which is causing the spoilage. And all these factors are governed by the environmental factors. Looking at the different aspects, how a biocontrol agent is interacting with the phytopathogen, it is combating the phytopathogen by different approaches, starting from the direct combat parasitism to the production of antibiotics, to siderophore production of volatile organic compounds, and the biocontrol agent in case of your host is interacting by inducing the induced systemic resistance mechanism. The biocontrol agent, it is interacting, especially when we talk about the post-harvest disease management, the faster rate of multiplication and growth of the biocontrol agent to cover the wounded surfaces or the intact surfaces will give a direct stress to the pathogen and deprive it from any chances of germination and further growth, and thereby protecting the concerned fruit and vegetable which we want to manage the disease of. And the desirable characters for the post-harvest biocontrol agents, as I said, that it should be having a very good capacity to grow, colonize the fresh wounds. The level of survival and rate of growth of the biocontrol agent should be good, and it should be able to withstand the post-harvest physical and chemical treatments. Very less work is done in order to understand that how the biocontrol agents which we are applying, they are interacting or they are withstanding the different physical and biochemical treatments. Talking about this aspect, especially different microbes are used, starting from bacteria like Bacillus, Anthoia, and mostly yeasts are having a very good important role in the management of post-harvest diseases. And one of the properties of yeast is that they can grow very fast and they do not produce mycotoxins. They are able to withstand the extreme conditions of low temperature, high temperature treatment given to the fruits and vegetables. They're able to withstand desiccation conditions. And all these factors make them a very good agent for use in commercial purposes for the treatment of fruits and vegetables and prevent the losses. I'm sorry. Here, this is a small picture where I have taken it from a reference paper, which clearly shows that what are the effects of the treatment with a particular biocontrol agent. The fruits are well preserved even after inoculating them with the pathogen, that is in case of citrus penicillium, and the other one, they are fresh and not affected by the pathogen, while in the other case where treatment has not been done, the fruits are showing the disease symptoms. And this is a small graph which shows that ultimately whatever product we are going to develop, whatever potential biocontrol agents are there, we must move towards the formulation of them. We must move towards the commercialization. When we talk about the formulations, then two types of formulations are popular: the dry formulations and the liquid formulations. Dry formulations have their own advantages. They are cheaper to be made and yeah, easy to be stored. But liquid formulations have their own advantages, like they're having a better shelf life. Whatever product we're developing, the consumers, they are always wanting for a product which is having a good shelf life. Different factors also contribute to increasing the shelf life. Like different agents are used, like your glycerol is used, ascorbic acid is used in order to improve the shelf life of the biocontrol agent in question. And lastly, this is a small flow chart wherein how the product development comes into phase, how we have to identify the problem, the crop in which we want to do the management practices, why is it important, what is the demand of the industry, what is the demand of the market. Depending on that, we have to pick up a problem, and we have to isolate the microbes. The microbes can be isolated by fruit washings from the surfaces. Dr. Jayanta Saha is here, and he's a pioneer, having great amount of work in this. I'm presenting my little bit of knowledge in this area, sir. It's privileged. And therefore, after the discovery selection process, you have to go for the bioassays. Bioassays are done in laboratory conditions. Thereafter, they are done in the commercial warehouses to see how the potential biocontrol agents are performing. Thereafter, we have to come to the formulation part. As I told, we have the dry and the liquid ones. They have their own advantages and all. Finally, there are some good biocontrol products which were introduced and still they are being used. Most of them are having a yeast as its active ingredient. And other than yeast, some bacteria like Bacillus, Pantoia are also favored. Thank you, sir.
Thank you, Ankita. Yeah, who is your guide?
Sir, actually, I have worked with Professor Asha Sinha and Professor B.K. Sarma. This is not my core work, but I found this topic to be interesting, so I picked it up.
Yeah.
I went through some review papers and I compiled it.
Yes, Tawad Alma Bhutia is there.
Yes, sir.
Yeah.
She's
She
here.
is there in BHU, right?
Yes, sir. Yes.
And what aspect she is working? She is working, still working with East.
Yes, sir. Yes, sir. She's working on East aspects. Yes, sir.
Nice. But it's a just gathering of information that you are
Yes, sir.
presenting.
Yes.
And lot of things are there for formulation. That's the most important part of Ashera. Lot of things has to be considered. Number one is not only the post-harvest, the storage environment, but also the yeast must have the osmotic stress tolerance capacity.
Yes, sir. Yes, sir.
The first one for formulation. If you go for liquid formulation, if you cannot maintain the osmotic balance cell inside the cell, each cell, as outside the medium that is having the formulation in the formulation, it will not work. Its shelf life will be low, very low. And another thing, the oxidative stress tolerance. And because the post-harvest diseases, so either it is quiescent infection or the infection comes during harvest or after harvest. That is also important. And while there is infection, there is oxidative fast. And if you apply the HG or any biocontrol agent, if we cannot tolerate the oxidative stress that is created in the field or in the harvested fruit, then again it will not work. These things, these two particular criteria must have to be considered while formulation. Okay, go on, but your main work is on different aspect.
Yes. Yes.
If you are interested, then keep contact with Tawad Alma Bhutia.
Yes, sir.
So that you can proceed with that.
Yes,
My tenure is complete almost. Only a few students are left yet. to be completed. Their PhD. And you people will carry out this work in India. I
Thank
am
you, sir.
trying to level.
Thank you for your blessings, sir. Thank you for your blessings.
Anybody, any question?
Okay, Ankita,
Yes, sir.
you have to go through literature and find out the first generation of biocontrol product registered and available commercially.
Yes, sir.
Whether it is available commercially in India.
No, sir, this is not. I don't know about the Indian aspect, sir. I took it out from a review paper. There, they have enlisted few of them. So,
No, in India there is no product of yeast. No product is registered. I heard that some company is trying to come up with a yeast-based product for post-harvest disease. But in India there is none.
So, what about the management of post-harvest diseases by this biocontrol product use?
Ankita,
yes, sir.
try to answer this question.
Sir, can you please repeat? I couldn't get your question.
What is the possibility to control or manage the post-harvest disease through these fungal or any biocontrol agent product?
Sir, these products, these biocontrol agents, they are giving a good response when we are going for the lab assays.
Ankita, one thing you should clear. One is the biocontrol agent, and second thing the biocontrol agent product.
Yes, sir.
So we are talking
Yes,
about the biocontrol agent product here only.
Yes, sir. So regarding the product, there are some bottlenecks. That is the problem. Even we are trying to develop some good formulations, although for a different disease and a different crop. But we are facing some bottlenecks regarding the osmotic stress tolerance and regarding the reactive oxygen stress, which is being developed in the formulated products. So it is a long journey, sir, and I think contributors from different places.
You mean that these products are facing the problem with osmotic and stress?
So not only that, because of this, there is shelf life we cannot guarantee. It is not giving a consistent performance in all the conditions.
No, you are talking about the biocontrol agent or biocontrol agent product?
I'm talking about the products.
Okay, where you have read this one? That product also suffering with the osmotic and stress problem.
Product is having the problem of not giving the consistent results. I mean, we are going to the farmers with the different biocontrol products of crop diseases. So they are they're always complaining about this aspect.
I think you are not getting my question. Actually, there are the two things. One is the biocontrol agent. That means biocontrol or fungal or bacteria as a whole, living organism. Second thing is the
product.
Any of the product,
yes,
other than the living organism. Is it or not?
Yes, sir.
You are written here, na, Aspire, Ecogen.
Yes,
these
sir.
are the chemicals. Actually.
Yes, sir.
These are chemical, not living organism.
No, sir. Yes, sir. They are the product. They are having the biocontrol agent and the inert materials and the adjuvants. Everything is added.
Want to input something?
Yeah. I'm giving you the input. This Aspire or Ecogen, this Shimar or Yield Plus. These all having live biocontrol agent.
Okay, live biocontrol agent.
Live biocontrol, yes,
mostly
sir,
the yeast. Yes. And out of these products, only few are there in the market, in the European market, particularly European is there. And the best.
Okay. That
the last name, the Shimar, the Mesnicoia fructicola. That is developed by Dr. Shomir Drabi from Israel. Now, still it is more than 8 years. It is still in the market and performing very well. And that is, I said, that is the important thing for getting a product with live organism, particularly the yeast. That is first thing we have to consider for development of the formulation. That is the osmotic stress tolerance capacity. Otherwise, we will not get the shelf life, good shelf life of that product, formulated product. And another thing, after application on the fruit, after harvest, then it will encounter the reactive oxygen due to injury or pathogen infection. If the yeast, the biocontrol agent, the microbe, if it cannot tolerate that oxidative stress, then again it's a problem. Having the good CFU count in your formulation, but it will not work if it does not have the oxidative stress tolerance capacity. These two things are very important and it will take time. Not only that, another aspect is there, microbiome, where it is going. There are several microorganisms that they are naturally coming on the fruit. Then these yeast or any living biocontrol agent, they will come across the competition between these. And that is also, and now it's now presently people are working in some other countries, particularly Israel, on this microbiome. Okay, okay.
Okay, thank you, Ankita. You can unshare your slide.
Thank you, sir.
And sir, one message come from Afroz because he is not in our list.
Afroz is not.
Session.
Yes, sir. My presentation was scheduled after 2.
Okay,
Oh.
okay.
Okay.
No,
Just
So
listening.
you are in the next technical session.
Yes, sir.
Technical session 3.
Anybody else in these? MS Mathi. So I think Dr. Shah, there is no more participant for presentation.
yeah.
So can we conclude it or we wait for some time?
What is Dr.
I think we can conclude till now. 146 is there.
146. Now within 15 minutes, I don't think that it will be completed. If anybody comes, what Dr.
5 minutes.
Bagh is saying? Yeah, 5 minutes. We can wait for 5 minutes.
Okay.
Then, what Dr. Bagh is saying? Dr. Bagh. Dr. Bagh.
Dr.
Have,
Bhatt.
you have completed.
No, we have completed.
Dr. Jadhav,
Yes, sir.
I request Dr. Jadhav to thanks all of you. Dr. Jadhav will present. Thanks.
Chairman. Dr. Shah,
Yes, sir.
could we conclude this? And I propose vote of thanks.
We should conclude. Because, how long
Sir.
we can wait? Yeah.
So one request, one participant was there. I have just informed him. I think he will just present. I have just called him, sir.
Okay, okay. Let him come very fast.
Yes, sir.
very
Yes, sir.
fast.
Okay, sir. Yes, sir.
Actually, time limit is too longer, na?
Yes, sir. Too long. Too long.
Though this is not too long, but as per the attendance of the
Yeah.
center.
And probably what I've seen in person symposium, seminar, or conference, what you will find, those who are participating in poster presentation session, they
But sir, one thing I could not understand.
Yeah.
Most of the participants written their local places like Bapatla,
Yeah, yeah,
Mandya,
yeah.
Karnataka. They have written, but as per the international conference rule, there should
Yeah,
be
yeah, it should be,
written
yes.
the country name.
And the country name and the pin code.
Yeah.
Pin. Yeah, it must be. I don't know whether it is there in the detail in their abstract or registration form or not, but it should be. And India, at last, India should be mentioned. Mm.
Sir, could we put on the chat box that in the next session, all the participants of this session 3 link and
No, session 3 chaired by different people.
I think next session chaired by some other fellow or
Other person. I have to chair only this poster, pre-harvest practice for quality and poster.
okay.
We have to leave by 2 o'clock sharp.
Ankita.
Ankita,
Yes, sir.
can you call him? What is
Sir, I have called him, sir. I have just called him. I just again call him 1 second, sir.
So it has over.
Excuse me, sir. Sir, one of my poster is yet pending.
How are you so late?
May I know your name?
What is your good name?
Jay Prakash Singh.
So where you were?
Jai Prakash Singh.
Now where you were?
2 बजे का इंतजार कर रहा हूँ.
Jayapaka say, what is
Okay,
the
you
poster?
you should start now.
No. You see first thing what is the poster number and session you ask? It may be a 3A or 3B.
3A,
Please
No,
count to
2A, 2A.
It was one from 1 to
2A.
2.
2A, 8 number.
This is 2A.
Yeah, 8. Yeah. Mr. Jayaprakash,
Share your
Yes, sir.
slide.
start your
Start slide sharing.
share your poster
Okay, sir.
very fast. We have limited time.
So it was all about quality deterioration in stored food grains owing to inappropriate moisture content and due to lack of aeration. Here, there are 4 objectives to find out whether the temperature, moisture content, humidity, and atmosphere were monitored. Second one is fungal genera were isolated from infested food grains. And third one is aerated and non-aerated chambers that were created for storage properties. And fourth one is presence of insect pest in godowns. Simple methods were used for fungal isolation, and different samples were isolated, from which 15 grains were separated and used on PDA plates for incubated for 3 days. Then they were separately grown for their culture characteristic, their colony color, and microscopic view. Further, in second objective, aerated and non-aerated chambers were maintained for checking their relative humidity as well as moisture content. So we observed the monthly moisture content, monthly relative humidity for the storage godowns. In the month of January, we observed that there were 13% moisture, and it was actually receipt started from April month. Wheat receipt started during April month. So the moisture was 11%, and after that is what slowly increased due to rain or high humidity. So it increased from 11 to 12, 13, 14, 15, 16 in non-aerated chambers. But in aerated chambers, it was increased only up to 13.4%. And it was directly proportional to relative humidity of the atmosphere. Whereas in case of rice, the moisture content was not much disturbed because moisture content in rice, as the rice grains are non-living. It is supposed that rice grains are non-living, but wheat are living as they respire. So moisture content of rice was, once it was received in the month of November, it was 14.9%. But slowly it decreased 14.9 to 8, 14.8, 7, then 14.4, and last was 13.7. It was never increased. Always losses their humidity and thus it lost their moisture content. At the end, it was observed that the major fungal diseases were Aspergillus, Flavus, Niger, Candidatus, Penicillium, Fusarium, Alternaria, and Mucor. And maximum fungal activity was noticed at 28 to 30 degrees Celsius, while relative humidity was 25%.
Jesse,
Moisture content, yes, sir.
you are telling that Aspergillus flavus, Aspergillus niger, Penicillium, these are the disease.
These are the major fungal genera isolated.
Fungal genera, so not from major disease of fungal disease.
No, sir. These are
They are just mycoflora of seed.
Microflora, seed microflora, yes.
And when the moisture content exceeded from 18%, then bacteria was also observed. I did not isolate septal bacteria, but it was noticed like a slime-like growth. And moisture content of grain and relative humidity of environment are complementary to each other, whereas moisture content regulates the biological activity of food grains, thus regulate the activity of insects as well as mycoflora. But in case of aerated chambers, the grain temperature, moisture content, and biological activity was maintained. Whereas in non-aerated chambers, there was huge humidity and with high moisture content, are increased insect population, increased fungal population. In non-aerated chambers, saucits were also found. That were the major indicators that that tells us that high moisture and blocked aeration results in insect population increase. So the saucits are the major indicators.
What is in the table? 14.8, 14.7. What is this?
Sir, this is the moisture content in aerated chamber and non-aerated chamber. MC means moisture content and RH means relative humidity of that month.
So non-aerated has the more moisture content.
Yes, sir. In non-aerated chambers, there was non-flow of air.
Okay.
So the moisture.
What is the initial moisture content of these seeds?
So, actually, wheat is procured in the month of April. So the moisture content.
It's okay. You can start any experiment scientifically, or you just start any way. Initially, what is the moisture content in aerated chamber or non-aerated chamber? Then, after some time longer, what is the?
Yes, sir. In the month of April, moisture content was 11%.
Just you listen here. You are written here in January 16% or what is this?
It's
in
non-aerated
chamber.
So in non-aerated chamber, procurement was moisture content was 11%.
Moisture content.
11%.
This is 16% in wheat. In January.
In January, it is 16%. Yes, sir.
16%. Then it reduced.
Then it's reduced.
Reduced. Then again it.
No, sir. It's the starting month. April is the starting month.
Starting month. Then it reduced. Then again high in the December.
Yes, sir. Yes, sir.
How? If there is no aeration, no water, nothing there, then how it can increase?
So because there is no aeration and relative humidity is high in the month of January, February, and March. But in the month of June, April, May, June, relative humidity is slightly low, like 75%.
In case of non-aerated chamber, how relative humidity can affect? I mean that. In case of the aerated chamber, it's okay. Relative humidity may be variable. But in case of the non-aerated chamber, how relative humidity can affect the
in non-aerated chambers, it's not completely blocked chambers. It is slightly non-aerated. Not it is completely blocked.
Okay, thank you. Complete your Dr. Saha.
Yeah.
Hello.
Jayaprakash, concluded?
also observed that in non-aerated chambers having damp condition due to high moisture and enhanced mold growth, which increased temperature from 35 to 50 degrees Celsius, as well as moisture content from 15 to 18%, known as wet heating. But in case of highly infested grains that were weevilled, it
But
means it was
Jai Singh,
yes, sir. Yes,
one
sir.
thing I do not understand, in January, aerated chamber and non-aerated chamber, both sample are same. 13
Yes, sir.
degree, 14.8 degree, 16 degree, 14.8 degree. How
Yes,
it is
sir.
different in the same month? Because it is the
Sir,
initial month.
sir, it is rice. Rice is procured at the
Okay, in rice, you can see in the aerated chamber, 13% moisture content wheat in aerated chamber,
Sir,
16% in non-aerated chamber.
the rice is received in the month of November, and its average moisture content is nearly 15%, 14.8, 13. It is approx moisture content. And wheat is received,
Not too much longer.
sir, in wheat, it is received at below 12% content. तो मतलब जब wheat का procurement होता है तो वो 12 से कम पे होता है और राइस का procurement 15 से
मैं
कम पे
वही
होता
तो
है.
कह रहा हूँ कि जब प्रोक्योरमेंट एक ही टाइम में कराया तुमने बीट का, बस वो रखे ही तो दो कंडीशन में, एरेटेड और नॉन एरेटेड में.
जी.
तो फिर उनका मॉइस्चर कंटेंट अलग अलग कैसे हो गया एक ही टाइम में?
सर
जनवरी
एक्चुअली
में तुमने खरीदा रखा कोई चीज, तो जनवरी में
Yes.
एक सैंपल लिया, दो जगह कर दिया, एक एरेटेड में एक नॉन एरेटेड में,
जी जी.
तो फिर कैसे कंटेंट अलग अलग हो गया?
Last one.
सर अप्रैल में तो सेम है ना सर, जिस टाइम
Dr.
हमने
Shah.
रिसीव किया.
Please you
Yeah.
stop it because another
Yeah.
presenter is there in this session. You stop it.
Yeah.
Close it. Okay.
Yes.
Fatima is there. Fatima is there. Again, you see the time.
Okay, leave it. Then thank you, Dr. Jaiprakash Singh.
Okay, sir. Sir, due to lack of time, I am not make to understand it. You all. Sorry,
Okay.
sir.
Yeah.
Okay, thank you.
This is so late. So late. Who is there? Fatima.
Thank you. Sir.
Fatima is there.
Sabah Fatima.
Is somebody else there?
Hello. Hello.
Hello.
Hello, sir.
Yeah.
Hello. Good afternoon, sir. Am I audible to you?
Jesse, unshare your slides.
Okay, sir. Okay, sir.
Hello.
Hello.
Yes, sir. Am I audible to you?
Yeah. You are Sabah Fatima.
Yes, sir. Sabah Fatima from Aligarh Muslim University.
Share quickly. Share it.
MS यादव सही कह रहे थे, ये ना लंबा खिंच गया बिना बात के, लोग कम.
क्या बताएंगे, हम लोग बैठा हुआ है जैसा एक होने के लिए, लेकिन कोई नहीं आ रहा. अभी 2 PM से हमका थर्ड सेशन शुरू करना है, इसके बाद आ रहा है.
ये साबा प्रतिमा कहाँ पर लिस्टेड है इस सेशन में?
इसमें है
Yeah.
12
Number
नंबर,
12.
12 number.
12 नंबर,
12 number in which session?
2A.
2A.
ये तो 3 बजे तक चलेगा फिर.
नहीं ये लास्ट है, इसके बाद तो कोई नहीं है.
This is last. You don't allow anybody.
Unshare Fatima, quick.
Okay, sir. Excuse
Okay,
me.
sir.
Can I ask one thing? Is this the technical section, poster presentation section of 3A?
No.
2A.
2A.
Your name is here. You should share your slide and start presentation.
Session 3A is not started yet. Okay.
Okay, sir. May I start now?
Yes.
Yeah.
Start.
Respected teachers and all my friends, a very good noon to all of you. Today, I, Sabah Fatima, going to presenting my research work through this poster presentation. So the objective of my research is applications of some weeds to test their nematicidal.
Sabah, you are not sharing your slide or poster. You are sharing yourself. Screen share is there, so you go.
Wait.
Okay, nice slide.
You first open PowerPoint mode, then you come to the here, you share, the security, participant, then chat, there is in green color, share screen, green color, lower portion you see.
First you open your poster on screen, then start sharing. It will appear on the screen. Dr. Saha,
Sir, it's
you tell me your mobile number. We can discuss later on after the session.
9433
9433
164
164
218.
218.
Yeah. Another one. This is
Yeah.
BSNL 700
700
3398
3398
185.
185.
Yeah.
Okay. We can discuss after this technical session because
Yeah. You have to leave this one. Yeah, this one.
Okay. Yeah, it starts.
Sir, there is no sharing option.
Sharing option, you see the
It is
lower
there at the
portion,
bottom.
In the bottom, share screen is there.
green color, you see green color,
Green
share
color
screen.
share screen.
Yeah, click on this.
Okay.
We have microphone symbol.
Now it has come.
Yes.
Now
It's
now
okay
you
now?
Is it
go,
okay? Is it okay, sir?
Yeah.
now you go in power presentation mode, you click there.
Okay. Sir.
If you don't get it, you start with this.
May I start from the beginning? Or should I?
Yes, you start.
Thank you, sir. So, respected teachers and all my friends, I, Sabah Fatma, going to presenting my research work through this poster presentation. And the objective of my research is applications of some weights to test their
You can
nematicide.
enhance the size of this poster by clicking on the plus sign.
Yes.
Yes.
So, applications of some weights to test their nematicidal behavior against Milidoga javanica under in vitro and greenhouse conditions damaging carrot. So in methodology for in vitro testing, the aqueous extracts of 6 different weights were taken in 5 different concentration, that is 20%, 40%, 60%, 80%, and 100% concentration. And for post experiments, 30 gram of chopped leaves of each weeds were taken, were combined in 5 gram dry leaf powder of Lapidium didymium as an organic amendment. So the data analysis of my results showed that the highest egg hatching inhibition was found in the aqueous extract concentration of Lapidium didymium and Malvacetum tricuspidatum after 7 days duration in 100% concentration, and whereas the greatest mortality of Milidoga javanica was recorded in the aqueous extracts of Lapidium didymium and Sonchus asper in 100% concentration at 72 hours of exposure period. Here we can see that 80% of hatching inhibition can be seen in 100% concentration of aqueous extracts of Lapidium didymium. So this table depicts that the combined treatment of Sonchus asper and Lapidium didymium is not only successfully reduce the population of Milidoga javanica, but also promote the growth of the plant in terms of shoot and root length, shoot and root fresh and dry weight. It was concluded that the aqueous extracts of Lapidium didymium and Sonchus asper has the highest effectivity against Milidoga javanica, and these two weeds when applied in as an soil amendment, are very efficient to minimize the population of root knot nematode Villedogine javanica. So we can use these to Lepidium didymium and Sonchus asper as a management tool to manage root knot nematode in future. So that is all about my poster presentation. Thank you all.
Okay. So Fatima, where you got this Lepidium didymium?
Lepidium didymium, I got it from my campus, my Aligarh Muslim University campus.
It is sufficiently available?
Yes, it is very sufficient, and it is grown commonly in March and Feb.
Okay. What about this Sonchus asper?
Sonchus asper belongs to the family Asteraceae, and it is also grown commonly, and it is also very sufficient in amount. So we can use this.
So what is the size of this plant?
The size of the plant is not so big. It is small in size. It's like a small shrub or a herb.
Okay. Thank you. Okay, Raksa.
Thank you so much.
So,
Yeah.
hello.
Hello.
I have one talk. No, it is not with Sabha. Dr. Birendra Kumar, you just wait. You are for next session. Next session is not started yet. Okay. And another thing here,
Oh,
Rajneesh Thakur is
okay,
present.
Dr. Saab, good morning.
Good morning.
Okay.
Hey, good afternoon.
Good afternoon. Okay.
Yeah.
Thanks.
You see, Rajneesh Thakur is present here in this session. Rajesh Thakur is agreed or not? You just ask.
First, civil.
Yeah. We
slide.
should.
Yeah, Rajneesh Thakur is there. In session 2A, third number, fourth number.
You ask he is agree or not. He want to present or not.
May I leave now, sir?
Yes. You answer.
You can answer.
Thank you, sir.
No, Rajneesh Thakur, one poster is in yesterday session at 0.7.
No, that is different name here. Here it is there.
So it's last now or Ankita Sarkar asking for someone?
Yeah. He presented. Jayaprakash.
Okay.
And now that Rajneesh Thakur is there or not?
Sabha, you unshare your
Yeah.
screen.
Dr. Lakshman, what is about next? There is no presentation in the list.
Rajneesh Thakur want to present or not? He is not responding at all. Yes or not?
No, he is not responding.
Dr.
Okay,
God,
you close.
Rajneesh Thakur is not in the list of this session 2.
No, he is the list in 2A at 4 number.
2A, number 4. 2A, number 4. Building of resistance.
Sabha, you
Yeah.
unshare your screen. Go on the top. Stop share.
Rajneesh Thakur, are you?
Sabha? Sabha Fatima. Unshare. No, go on the top of your screen, then there the stop share. You push that button only.
Or you leave, or you click the leave. It will go. If you cannot.
You can leave.
You can go on the leave choice.
Admin can disconnect his Sabhapati last.
Cannot be no.
Sabha Fatima, you can click on leave button.
Here in the right side.
Right side in the bottom. Right side on
Right
the
side bottom.
leave button.
Right side button.
Right side bottom,
The
right
bottom.
side bottom.
Yes, yes.
So
Lakshman, you see that Rajesh Thakur is not responding. You close.
yeah, yeah.
Yeah.
Now we should conclude as because the other session that will be delayed.
Yes.
It should not be in online mode. It should not.
So, sir, we and Dr. Saha will discuss about the all presentation and send report to you.
Yeah.
Yes.
Due to the shortage of time, we cannot discuss here.
Yes, of course.
Yeah. Then Dr. Bag.
Yes.
Yeah. Thank you very much.
We are highly thankful to Dr. Jayant Shah and Dr. Lakshman Prasad for nicely conducting this session. And we are also grateful for their exhaustive task, and they have kept so much patience, and due to their hard patience, 4 more presentation was possible and opportunity for scientists was given. So we are highly thankful to you both for such a commendable job you have did for 3 hours continuously. So we are highly thankful to you both, sir. And I am also thankful to Dr. T.K. Bag for keeping patience and conducting, organizing this session. Thank you very much to all.
Thank you, Dr. Jadhav.
Thank you, Dr.
Thank you, Dr. Saha and Dr. Lakshman Prasad. You see that we started at appropriate time, okay, at 11. But in the middle, candidates are not there.
Yeah.
We, wait for them.
Any but percentage will increase later on.
Chairman of the session 3 is also on the line, and he is waiting for starting.
Now we should leave.
Kajal Viswas.
Okay, thank you everyone.
Okay, thank you,
Okay, thank you, sir.
Thank you.
Sir, can I call you after 1 hour?
No problem.
Okay.
Okay, thank you.
We'll discuss, and by the evening I will send the report and a copy to your Dr. Prasad. Your mail address will give me.
We can discuss and prepare the report. Okay.
Okay.
Okay, thank you.
Yeah.
হ্যালো জয়ন্ত দা, কেমন আছো?
আরে বাবা, দেখি রে।
Kajal is Dr. Kajal.
I am for the next session because I am driving my car and I am in my mobile, and after reaching to office, then I will put on my computer.
Okay, you come.
Tushar, you can start.
Yes.
Because otherwise it will be late.
Yes.
Now you are not driving. Now I am not talking with you. You just go.
Okay, I will talk evening, Dada.
Yeah. Fine.
Okay. Fine.
Okay, thank you.
Thank you.
Dr. Viswas, we are starting session.
Yes, you start.
Okay, come on the office.
You start. Don't worry.
Now we are going to start the second session.
Third session.
Third session, that is 3A, and after that 3B will be there. Okay, and here is the session 3A. There is chairman is Dr. K.K. Viswas and co-chairman is Dr. Virendra Kumar. So let me introduce Dr. Viswas. Dr. Viswas is a renowned plant virologist. He is working here in the division of plant pathology. He is having about 25-year experience in research, teaching, and extension. He is fellow of Indian Phytopathological Society, Indian Virological Society, and Indian Society of Mycology and Plant Pathology, Udaipur. And he has several awards in national reports. Professor M.J. Narasimhan Academy Award given by Indian Phytopathological Society, and SPPS Meritorious Scientist Award, Society of Plant Protection Science, and Sikha Ratan Puraskar, India International Friendship Society. He also got Best Paper Award of IPS KPD Menon Best Poster Award from the Society, and PP Singhal Memorial PI Industry Award from the Society of Mycology and Plant Pathology Udaipur, and recently got SB Chattopadhyay Memorial Award from Indian Mycological Society Kolkata, and JF Dastur Memorial Award Indian Phytological Society, both of these in 2020. He is having about 123 published research articles in national and international journal, and published 2 books and several externally projects funded by DBT, DST, and ICR. And with this, I welcome Dr. Kajal Kumar Biswas to chair this session for 3A first and next 3B. Next co-chairman is Dr. Virendra Kumar. He is presently Professor cum Chief Scientist from Department of Plant Pathology, Tirhat College of Agriculture, Bihar. He also handled several projects, 6 projects handled, and co-principal investigator of several projects, and guided MSc and PhD student. I welcome Dr. Virendra Kumar as a co-chairman in this 3A poster session. So now I request chairman and co-chairman to conduct this poster session presentation. Dr. Biswas and Dr. Virendra Kumar, I request you to take care this session.
That's all. May I submit one thing?
Yes.
There are chat question on the chat that what is the link of this session. So once you have told and again repeat that, same link will be work in for all posters.
Yeah.
So all the presenter of this session 3A link as soon as possible.
Yes, 3A and 3B both will be here in this link.
Yes, sir. Okay, sir.
Okay. So Dr. Virendra Kumar ji and Dr. Biswas start this program.
Because Dr. Biswas is on the way, so Dr. Virendra Kumar can initiate the session.
No, I reached. No problem. We start. Dr. Virendra Kumar. Dr. Virendra Kumar, please start. I am always.
Okay, sir. Good afternoon.
Virendra Kumar.
Okay, sir. Good afternoon, sir. Just start a poster session. Hello, sir.
Yes.
Okay, sir. So first of all, I thank to all organizers to nominate me as a chairperson of this poster session. Hello, sir.
Yes, yes.
Yes. So some actually problem in net. Already I told him.
No,
Okay.
your sound is also audible and your picture is also coming.
Okay, so
Okay, no
sir,
problem.
in this session, poster session 3A,
R and Sharma started. Number 1 is R and Sharma.
Okay, so you start R.N. Sharma.
What is the time, Dr. Tushar?
Hi.
2 minutes for presentation.
2 to 3 minutes.
Any question?
Yes, you do question.
Chairman has a power to question.
Okay.
Dr. Biswas, good
Yes.
afternoon. Good
I have
afternoon,
seen you after a long time.
Oh, okay. When you come to
Because
Pusa?
2016. I hope so.
16,
After that, we
okay.
could not get
Okay.
opportunity to meet you.
But yesterday I tried to call him, but they do not have any mobile number. So I'm sorry.
Actually, Shubhankar joined. Now, after that, I am thinking I will go there, but I could not go. Could not able to go.
So I request you chair the session. I also along with
No
you.
problem. We are both are same.
Actually, 3 p.m. one another meeting, so 1 hour we take time.
Okay, I will be here. No problem.
Okay, so you start, no?
Okay, you start. Now you announce the name of R.N. Sharma for presentation of his poster, biocontrol of post-harvest penicillium rot of Kinno Mandarin, I hope so. No?
Yes, Kinno.
Yes.
The Kinno Mandarin, what is the Kinno Mandarin? That is the Deliciosa. No? Okay. Please.
Okay, so R.N. Sharma, you start.
He has not linked up to now.
So if he is not there, you move to second one.
Another is Marabonia, Sandhya Rani.
Why not these presenter are got linked?
Dr. Bagh, first you announce a lot who are the present.
Hello, sir. I am in poster number 3, Dr. M. Prima Devi. I'm present.
So you start presentation.
Okay,
yes.
Okay.
Yes, you share.
Prema not visible.
Yeah, because I'm having some problem with sharing.
Okay.
They're saying only PDF, no, only for this one document. Okay, let me just click a photo and.
First you click on share screen.
Yes, I have clicked.
First you open your PPT. Okay, then you click the share screen.
Yes, sir.
Okay, sir, is my slide visible to you?
No.
Now it is starting.
So now is it visible?
Yeah, now it is visible.
It is visible. Yes, you increase the size.
Now is it okay, sir?
But some portion is cut. It is increasing,
Let
but
me-
left and right both side is cut.
But we will continue, no problem, because I think this is Microsoft Word.
No, this is PowerPoint, sir. Actually.
Now it is coming. Now it is coming. Coming.
Start.
Okay. So, good afternoon, everyone. Respected chairman and every participant present today. Myself, I'm Dr. M. Prima Devi, assistant professor in the Department of Pomology and Post-Harvest Technology, Uttar Banga Krishi Vishwavidyalaya, West Bengal. So today I'm here to present a poster on effect of pre-drying fungicidal treatment on storage life of dry spray chrysanthemum. So this was one of our research work. I would like to emphasize, since I got only 2 minutes, so flower, it is mostly used for its aesthetic value, but the shelf life is very poor. So we have tried drying as a means to preserve the flower, but due to its highly hygroscopic nature, it became very difficult for the flowers to be stored, the storability of the flower to be extended. So because of the microbial infection, specifically the mole infection. So a research program was keeping that in view, we have conducted research work on the objective, first of all, to study the pre-drying treatment on quality and storability of dry chrysanthemum flower, and to see the effect of storage condition on the storage life and quality of dry chrysanthemum flower. So in this study, we have taken 2 contact fungicide, that is mancozeb and copper hydroxide at a rate of 2 gram per liter, and 2 systemic fungicide, hexaconazole and azoxystrobin at the rate of 1 ml per liter. They were stored in different packaging material. First of all, it's a polyethylene pouch, then vacuum packed airtight plastic jar with silica gel, and airtight plastic jar. Control was in open condition. So from this comprehensive research trial, I've observed that the application of, or we can say the pre-drying fungicidal treatment of chrysanthemum after drying and storing have a great impact. As you can see from the table that I have just given 2 tables because of the scarcity of space. After 15 days, observations were recorded after every 15 days interval. So up to the shelf life, up to how much it could be stored. So shelf life was decided as per the qualitative and quantitative parameters, as well as the microbial attack, microbial attack, and most importantly, the appeal, since flowers are used for its aesthetic value. So according to this criteria, maximum score for all these parameters, it was observed in azoxystrobin treated dry chrysanthemum flowers stored in plastic jar with silica gel. While minimum score was found in mancozeb treated dry flower stored in open condition. While talking about the shelf life, the shelf life was maximum in my study, it was 105 days. But fungicided treated dry chrysanthemum flower could also be stored for 105 days, irrespective of the treatment and storage condition. But the thing is that the qualitative property, that is the poor scoring, was observed in all the qualitative property, like the attractiveness, the color, texture, brittleness. It was having the least score. But the best treatment was found in terms of high shelf life, qualitative as well as quantitative parameter, and also lesser microbial count. So you can see from these pictures also that the dry chrysanthemum flowers, specifically the yellow one I have chosen, because even after storing for 105 days, it looks almost more or less 90% similar with the fresh one. So the affected dry flower were observed under microscope, and we have observed that Aspergillus and Rhizopus with different level of contamination at different time interval were observed. So among all the treatment, I would like to summarize that the best combination is azoxystrobin treated pre-drying treatment of azoxystrobin when stored in a plastic box with silica gel, recorded to have no contamination up to 105 days of storage. And the best treatment, second best treatment, was hexaconazole with vacuum packaging. So this was the finding which I have observed in this. Thank you, everyone.
Thank you, Prema. You are a faculty or you are student?
I'm a faculty.
You are faculty?
Yeah.
Yes. You belongs to which?
I belong to, sir, pomology and post-harvest. My discipline is post-harvest technology of horticulture.
Your home in the North Bengal?
No, sir. I'm actually from Manipur, but I'm working in North Bengal.
Yeah. Now, just one simple question I will ask you. Now, yes. The fungicides you are applying, so what is your target pathogen?
Sir, we have mostly in general sense, because I am not a pathologist. No. In general sense, I have done so many research work on drying, sir. So I found that mole growth is more. So I have consulted with one of our pathologists, S. Khalko, and he have suggested me this fungicides because the main problem was fungus.
Then which fungicides is the good for your this kinds of work?
Hexaconazole and systemic fungicide were better because we did both systemic and contact, and then while dipping it and then directly placing the flower head. So placing one was not giving a good result, and dipping the flower stalk before drying, that was giving a good result post drying during storage.
Okay. Thank you. Thank you. Huh?
Okay, sir. Thank you so
Okay.
much.
Now you
shared your slide.
Yeah.
Yeah, that's what I cannot find it right now. Okay. Just a moment.
Hello.
Stop your You say skin.
Unshared,
You stop the skin scare.
If nothing possible, then leave.
You leave, then again you join.
You leave in the bottom right side.
And then again you join. Then the next candidate can start.
And next candidate, serial number 2 is present. I saw.
Adabin Mara Boina.
Sandhya Rani, okay.
Now Sandharani, you share your screen.
Okay, sir. Sir?
Yes.
Sir, make me host, sir. Kindly make me host, sir.
Yeah, host karo usko. Who is operating? Jadhav sir. Accept karo unko.
They did not send to us earlier, no? We don't have.
No, already sees in here.
Yes, it is coming. You try. Yes.
Sir, is my screen visible, sir?
Yes, visible. You increase the size.
Okay, sir. Sir, is it okay, sir?
Yes, it is okay.
Good afternoon, everyone. Myself, Sandhya. My
And
topic is...
You decrease to some extent. So that we can see your whole slide.
You see that it is made in PDF.
Okay. Still reduce, no problem. Okay. No, it is more reduced. Now we start.
Okay, sir. Nanotechnology, advanced technique in post-harvest management of horticultural crops. Coming to introduction, the post-harvest losses estimates are 1/3 of food products every year, and it is one of the most important issues in the storage of fruits and vegetables. These losses may depend upon many factors like type of crop, method of crop harvesting, distance from field to storage, etc. The fruits and vegetables are perishable in nature and their chances to spoil are more. The major reasons for the post-harvest losses are microbial attack, moisture degradation, physical and chemical contamination. Nanotechnology has arrived a great help in all science and industry branches. Nanoparticles have particularly characteristics are useful in post-harvest technologies. It can improve the post-harvest shelf life by controlling the growth of different development and spread of disease-causing microorganisms. With the development of new generation of packaging coverings, that is films, increases strength, quality and packaging beauty, and uses the nano biosensors for labeling the food products has proved beneficial for controlling post-harvest losses. The major source of contaminants in post-harvest storages are the physical contaminants. The physical contaminants are foreign materials which come embedded inside fruit or vegetable, can cause a serious injury or illness if consumed. These contaminants include soil, wood, broken glass, plastic or metal parts like nails, nuts or bolts. Coming to microbial contaminants, this is one of the most common post-harvest pathogens around the globe. Botrytis cinerea, it is a fungal pathogen which causes grey mold disease. It is responsible for decay of economically important fruits and vegetables during the growth period and post-harvest storage. It also makes the long-distance transport and storage impractical. The pathogen attacks the plant either by direct penetration or by indirect entry via wounds or mechanical injuries, and can infect all the parts of plant parts like leaves, stems, flowers, and fruits. The fungi develop at low temperature conditions, minus 0.5 degrees centigrade, and spread rapidly. Hence, it is important to control during the storage. Coming to chemical contaminants, the fungicides that are used for microbial control can ameliorate the loss and ensure product protection. But these cannot be used for all the horticultural species. In addition, public awareness of the adverse effects of synthetic fungicide residues on the human health and the environment has prompted the controlled use of major chemicals. The major issue that prevails is that resistant development in pathogen against the synthetic fungicides. Coming to the role of nanotechnology in post-harvest management. Coatings. Coatings has proved to be effective in reducing losses in storage as it acts as a carrier of useful ingredients like antioxidants, antimicrobial compounds, anti-ripening compounds, color or aroma additives. It has many advantages such as retardation of ripening, reduction of water loss, chilling, mechanical injury, and decay. It also adds shine to the coated products. Different coating materials can be used like lipids, polysaccharides, proteins, or a combination of these materials. Coming to natural antimicrobial compound, chitosan can be used to lower the post-harvest decay of horticultural products. It is deacetylated from form of chitin, which is an eco-friendly means of pre- and post-harvest disease control. It has been reported that chitosan, being a natural compound, can successfully substitute the synthetic fungicides by improving resistance in tomato against grey mold caused by Botrytis cinerea. Coming to nanotechnology, has been utilized as a source of antifungal treatment in many fruits and vegetables. It has produced nanoparticles, zinc oxide using microbial approach, some of which has been used for post-harvest storage of banana, carrot, tomato, onion, etc. The introduction of a new generation of packaging cover films has been useful in protecting post-harvest produces. The mixture of the polymers with organic and inorganic additives of certain geometries, spheres, flakes, fibers, or particulates produce polymer composites. With the use of nanoscale fillers, polymer nanocomposites are developed that has become a radical substitute for the conventional polymer composites. The nanocomposites have proved to be successfully
please do fast because time is. You tell the important findings, what you have got in your research.
Sir, actually, this is
What
not
is the
my
conclusion.
Okay, sir.
Yeah.
Nanotechnology has proved to have high potential in improving the quality of horticultural products. It is advanced technique that uses different nanopacking materials to be utilized in post-harvest management, and it is more effective in controlling physiological and physiochemical quality in contrast with normal packaging material. Furthermore, these nanomaterials have simpler processing and industrial feasibility than other storages, and are less time-consuming, less costly, and do not alter color and flavor of the fruit and vegetable. Thank you, sir.
Thank you for your presentation. But what are the research you have done in this poster?
It is review paper.
This is a review paper. Okay, now you tell me which is the most important nanoparticles are used in India, and what the nano formulation are available commercially in India. Please tell me. What are the important nanoparticles?
Sir, titanium dioxide.
In plant pathology, which is the most important?
In plant pathology, natural antimicrobial compound chitosan.
No, which nanoparticles are mostly used in India?
Chitosan is mostly used, sir.
Silver, copper.
Chitosan. Titanium dioxide.
No. And what is the phytobiosynthesis of nanoparticles? Do you have any idea?
No, sir, I don't have any idea regarding that.
You are PhD second year or third year?
First year, sir.
First year?
Yes, sir.
Okay, that's why. Okay. And your thesis program on this nanotechnology?
No, sir, this is general topic I have taken, sir.
General topics, but what is your PhD topics? I thought, I'm thinking your topics will be on the nanotechnology. Okay, thank you very much. I would stop your screen share. Then we can call the next person. R.N. Sharma, he is present or not?
Now, here you see, I am informing you, number 10, Susa S. Tharar,
Yes, yes, sir. I am.
present here.
Susa Tharar.
Yes, yes, sir.
Okay,
Can I share, sir?
yes. In speaker view, always MS Jadhav is coming.
Yes, yes, sir. I think it's a little bit slow. Okay, sir. Can you see it, sir?
Yes, yes, it is coming.
Yes.
Earlier
Okay,
it was
sir.
good, it is too small. Earlier it was good.
Okay.
Yes, yes, it is okay.
Okay, sir. Okay, sir. I am Dr. Sushaya Tara, working as assistant professor in College of Agriculture, Kerala Agriculture University. And I'm doing with the post-harvest management of banana. And I go across a number of diseases. And banana, it is considered to be a globally important fruit crop. As far as India and Kerala is also concerned, it is very important in the homestead cultivation and all. And among the cultivation, it is affected by a number of pathogens. And especially in Robusta varieties, that is AAA, there's a post-harvest crown rot problem that will lead to the detachment of the fruit at the consumer level, or a problem at that side. And now the carbendazim that is been applied on the fruit surface in order to reduce this problem. But in our nationally accredited lab, there is the detectable amount of carbendazim is seen in the banana fruit, and we are considering about some biocontrol strategy. And survey was conducted throughout Kerala, and major pathogens. The main aim of the project is to develop an eco-friendly method for this. As a part of this, this is the screening of the antagonistic microbes for the management of the pathogen. And from the survey, the most important pathogens are found as Laccidiplodia theobrome and Colicotrichum musae. And the Laccidiplodia theobrome, as in the picture, we can see it causes the complete rotting of the fruit. Here you can see. That is the all fruit that is covered by a fluffy mass, and it will easily converted into the liquid mass. That is, liquefaction is major problem in this pathogen. And also another pathogen. So there is also another pathogen also found severe, that is Colicotrichum musae, that sometimes we can call it as anthracnose or anything. This also lead to the detachment of the fruit from its stalk, so that the consumer level, it will become a major problem. So as a part to manage this, as a number of organisms from the fructosphere, the different microorganisms are isolated out. And first of all, it is screened for its efficiency in the management of the major, these two pathogens, Laccidiplodia theobrome and as well as the Colicotrichum musae. And from the earlier screening was done for the pathogenicity, the organisms which are found to be non-pathogenic to the fruit, they are selected for the dual culture. And from that, there are 2 or 3 major appealing antagonistic organisms was selected out. And one is Aspergillus acuminatus and the Aspergillus niger, and some of the already established biocontrol agents like the Pseudomonas fluorescens, Trichoderma viride, Trichoderma halcyanum, Bacillus subtilis, and also the isolated and which are found to be promising in the dual culture are selected. And from this comparative study, it is found that this the Trichoderma harzianum that we can see, it is producing a clear-cut inhibition to the growth of the pathogen, and it is followed by the two native isolates of Aspergillus. They are the Aspergillus acquaintance and the Aspergillus niger that are you can see as the W3BC and A3BC. They are the two native biocontrol agents that are isolated. From the table itself, we can see the VESTA inhibition can be seen in the in vitro studies by the followed by the Aspergillus aculeatus and Aspergillus niger. And also like this, these organisms were also dried against the Colletotrichum musae. And here also the Trichoderma harzianum, it is having a higher inhibition rate, about 78% inhibition as seen by that. And the other two, the organism Aspergillus aculeatus and Aspergillus niger, they are also found to be effective in the in vitro studies. And from these in vitro studies, we found these organisms both are effective against both the organisms like the Lasiodiplodia theobrome and Colletotrichum musae, which are found to be a major post-harvest pathogen as far as the banana is concerned. Okay, thank you, sir.
Thank you for your nice presentation and very good poster. Now I have one query. What is the formulation you will be using for the control of the crown rot of banana?
Is it chemical or the biocontrol agent, sir, mean?
That is, you have screened and Trichoderma harziana.
Yeah, I have screened and actually this is the in vitro studies as a part of the project. We have also continued.
Yes. And now tell me how it applies.
The formulation we are using the 10⁻⁶ CFU per ml. It is found to be very effective, especially this Trichoderma harzianum as the post-treatment application and the pre-treatment application, so that we can prevent both the latent infection as well as the infection in the storage. It is found to be very effective. And when we are considering the temperature, okay, sir. When we are considering the temperature for the consider this Lasiodiplodia, this is a temperate loving. Even at 35 degrees Celsius, it is growing very fast. So that for the incubation period and all, this become a severe problem, especially through the cutting.
You have tested. Now you are going to test.
Yeah, I have tested, sir. We have an ongoing project on the state science and technology project.
You will show in your next paper. I hope so.
Okay, that will be in the sir. This is only a part, sir.
That means that you have made formulation. Now you are going to prepare the formulation.
Yeah, sir. After that, I have not finished it. Our continuing project is on the way.
Okay, Dr. Virendra Kumar, you have any question?
Okay, sir.
Okay.
Sir.
Thank you, Tara. Now please
sir, thank you.
Stop your screen, share screen.
Okay, thank you, sir.
Thank you.
Now serial number 16 is there.
16.
Sakshi Tomar.
Yes, sir, I am here only. So should I share it?
Yes.
Sir, it is showing that you cannot share start screen while the other participant is sharing. I think ma'am didn't leave yet.
Dr. Tara, just you stop your screen share. Otherwise, you leave and then again you join. Hello.
So she didn't left yet.
That's why you are not sharing.
Yes, sir.
Tara, Dr. Tara.
Yeah, now I can share it, sir.
Okay, now it's.
Okay, sir. Okay, it should be in. Is it fine, sir? Should I start?
You increase to some extent the size.
Wait a sec, sir. I'll try again. Sir, is it fine now?
Not coming. No. Yeah, please increase to some extent.
Sir, I have already increased it up to
Okay.
100%.
You start.
So, good afternoon, everybody. Myself, Sakshi Tomar, and on behalf of Dr. Dinesh Singh and Dr. Srinivas S, I would like to present my poster on the topic controlling pre and post harvest disease in mango using Trichoderma herzianum strain T2 bioformulation. So, as we all know that bioformulation are widely used as biological active products, which consist of singular consortia beneficial microorganisms or the microbial strain that we can use for controlling and eradication of diseases, as well as which will going to accelerate the growth of the crop as well. So in this particular studies, we aim to develop such a big broad spectrum bioformulation which we can use in both the pre and the post harvest disease control in mango. So we have isolated 8 different strains of Trichoderma from the Mango Orchard ICR IRI. And then out of that particular 8 strains, we have selected one potent strain, which was named as T2. And it was actually inoculated with 2 of the fungi. One was Fusarium mangiferae, which was causing the pre-harvest diseases in mango in the form of mango malformation. And another one was with Coletotrichum gliosporates, so which causes anthracnose in the post-harvest as the post-harvest disease in mango. So during the course of our studies, we have conducted the in vitro and both the farm studies as well as. So in the lab studies, we concluded that there is actually there is a particular property of this Trichoderma herzianum, which was inhibiting Fusarium mangiferae and Coletotrichum gliosporates. So after that, sir, we have moved to orchard, and then we have sprayed 100 gram of the bioformulation into 25 liters of water, and we have added 3 grams of jaggery in it. So jaggery served as an adjuvant. So when we have sprayed it on the tree, the mango malformation has decreased by more than 98%. And when we have actually treat the mango fruit with the same bioformulation, it was 20 gram in 100 ml of the solution. So after that, what we have discovered, there was a decline in anthracnose, and the decline rate was 90%. So in GCMS studies and SEM studies, which we have concluded, GCMS shows different antifungal and antibacterial compounds which were present in the bioformulation, which was made out of Trichoderma herzianum strain T2. And some of the compounds were 110 octadecanoic acid, 2,6-hexanoic acid. So these 2 were having the antifungal property. And similarly, when we have seen the scanning electron microscopy images, so what this fungi was actually doing, it was parasitizing, and it was actually attacking the another fungi, and it was creating anaerobic environment, which lead to the gradual death of the infectious fungi. So this is actually the best method or the natural method from which we can treat both pre and the post-harvest diseases in mango. And we have actually got the accession number of this particular strain, and we have already supplied it to farmers in Saharanpur, and they are actually using it at the moment, and it is actually revealing very good results. So we have applied for the patent as well, but it is in progress at the moment. So at last, I will be thankful to Dr. Rashmi Agarwal ma'am, who is the head of the department Division of Plant and Pathology, and Dr. K.K. Vinod sir, Dr. Gopalakrishnan sir, Principal Scientist, Division of Genetics, ICR IRI. And I would like to dedicate to my today's presentation to Dr. Srinivas S, whom we lost due to pandemic this time. Okay, sir. Thank you so much, sir.
Thank you for your presentation, Sakshi. Now I have
Yes,
one
sir.
question. one or two, three
Yes,
questions.
sir.
I hope so.
Yes, sir.
First
Yes,
thing,
sir.
how you have shown 98% lesser malformation
Yes, sir.
and 90% decline in the
Yes.
anthracnose. How you are applying this T2
Yes, sir.
Trichoderma?
Yes, sir. Yes, sir. We are
Where
only
you have
applying.
applied? Which orchard?
Sir, we have tested it in our orchard, the horticulture orchard, under the guidance of Dr. K. Usha ma'am.
Where? Which orchard?
Sir, in ICR IRI only.
Where that orchard
Yeah.
is located?
Sir, it is located just opposite to this floriculture
And
field.
how you have applied?
Sir, we are just applying it by the simple spraying method, and we are actually using 25 liters of the spray, in which 100 gram is of the bioformulation and 3 grams is your jaggery.
Yes.
And sir, we have given it to, since ma'am is having some farmers in her MGMG program, they are having orchard in Saharanpur, so we have conducted studies over there also, and in my orchard in Meerut also.
Yes. Now in the reference Dasgupta SN,
Yes, sir.
1993 in Lahore.
Yes, sir.
1993.
Sir, it was actually there only.
1993 SN Dasgupta. That is Dasgupta will be the together. Das, comma, Gupta, comma, SN.
No, sir, it is
What
actually
is this?
3
Lahore.
people. Yes, sir. Actually, my colleague Neha
It's
ma'am.
wrong.
Yes, sir. I will going to rectify it.
Okay. Okay.
Any more questions?
Dr. Virendra Kumar, any question you are having? Okay. Now stop your screen share.
Okay, sir. Thank you so much, everybody, and have a nice day.
Serial number 12 is present.
Deepa,
Yes, sir, I am present.
now please share your
Okay, sir.
poster.
Sir, is my poster visible?
Yes, yes.
Good afternoon, all respected teachers and my dear friends. Myself, Deepa R. Chandran, pursuing PhD in Department of Plant Pathology, College of Agriculture, Vellayanni, Kerala Agricultural University, Thiruvananthapuram. Today, I am here before you to present on my poster on the topic in vitro assessment of the inhibitory effects of essential oils on the major pathogen causing post-harvest crown rot of Robusta variety Musa AAA banana in Kerala. We all know that banana is an important fruit produced, consumed as well as exported in large quantities. And post-harvest losses in banana are very high, and majority is due to loss by post-harvest diseases, and of which more serious problems are crown rot and anthracnose. And crown rot is incited by a complex of fungi, and I am presenting a part of the study conducted in our department to identify the major pathogen associated with crown rot of Robusta variety banana in Kerala and develop eco-friendly and stable management strategy for its management. And use of synthetic chemical fungicides is still being the primary method of controlling post-harvest diseases, and drawbacks of chemical pesticides necessitated to develop alternate technologies. And coming to the objectives of my study, we is to survey the major banana producing areas of the state and collect the disease samples from farmers market, then to isolate and identify the major pathogen associated with crown rot of banana in Kerala, and to assess the inhibitory effects of essential oils on the major pathogen causing post-harvest crown rot of banana. And the materials and methods used, we surveyed the major banana growing areas of Kerala, that is Thiruvananthapuram, Alappuzha, Wayanad and Pathanamthitta districts, and the disease samples were collected, and we isolated the pathogens, and pathogenicity test was done, and virulence rating was also done. And effect of essential oils against major pathogen was studied by using the poison food technique suggested by the Nine and Taplial in 1993, and we used 4 different concentrations of the essential oils, namely basil oil, thyme oil, cinnamon oil, clove oil and virgin coconut oil, and the concentrations we used include 0.025, 0.05, 0.1, and 0.5% respectively. And you can see from the plates, first plates show the symptoms at 5 days after artificial inoculation of the isolates we got from different locations in the surveyed locations of Thiruvananthapuram, Alappuzha, Pathanamthitta, and Wayanad districts. And the second plate show the effect of essential oils, different essential oils on the growth of pathogen. And starting from the basil oil, thyme oil, cinnamon oil, clove oil, and virgin coconut oil along with the control at 4 different concentrations. And from the study, we could confirm that the major pathogen was the T2C isolate that is obtained from the Thiruvananthapuram district, and the molecular as well, the morphological as well as the molecular studies confirmed it to be Lasiodiplodia theobrome, and we have deposited it in the GenBank accession, and the accession number is shown here. And also you can see the growth of pathogen in the different concentrations of essential oils. And what we could infer from the experiment was that the growth was completely arrested at 0.5% in all the essential oils, and for 0.1% of concentration, the growth of the pathogen was arrested only in cinnamon oil, clove oil, and basil oil, whereas at 0.05%, the complete arrest of growth was in cinnamon oil. And coming to the 0.025%, none of the essential oils arrested the growth of the pathogen. And also you can see the aerial view and the cross-sectional view of peel of the treated as well as untreated banana. There you can see in the first section, it is the treated banana peel that it is with the cinnamon oil at 0.05%. There you can see there you can't see any infection or any development of the fruiting body of the pathogen. And I would like to say that we all know fruiting body of the Lasiodiplodia is a pycnidium. In the second picture, we can see the diseased or the untreated banana peel. There you can see the brown lesions. Actually, it is a microscopic view of the pycnidia. And what we could infer from the study, the part of the major study is that cinnamon oil at 0.05% has maximum inhibition at the lowest possible concentration. And the takeaway is that this can serve as an effective biocontrol strategy against the major pathogen Lasiodiplodia theobrome, which is the major crown rot pathogen of banana in Kerala. And with this, I would like to acknowledge Kerala Agricultural University for the facilities, funding, and the fellowship, as well as Kerala State Council for Science, Technology, and Environment for the fellowship. Thank you for giving me this opportunity. Have a nice day.
Thank you for your presentation. You are the PhD scholar?
Yes, sir.
You are working with Shusha?
Actually, sir, this was my MSc study, and she was my chairman.
Oh, in
I
MSc.
worked as a project fellow for that STEC funded project.
Yes. Now star is the Shusha is Taras, and the star is there. That means
Yeah.
and your star where not. That means I am not understanding. That's why I'm asking you.
Okay, sir.
Now, Dr. Shusha has worked on the crown rot. There's the polytrichum, and you are working on Lasidium. Acha, that is your MSc work.
Actually, sir, actually that was also done in my work only. The thing is that we got several pathogens, like more than 30 pathogens we got, but of that majority were Lasiodiplodia species. And when we rated the virulence also using the PDI and all, we got Lasiodiplodia species as the major pathogen. But Colletotrichum was the next best, means the second major pathogen. And except from our study, we could see that the Colletotrichum musae was the major crown rot pathogen. And our study only showed that it was the first study to show that Lasiodiplodia theobrome was the major crown rot pathogen in Kerala.
Yes. Now, cinnamon oil, how will be practically useful for the farmer?
Yes, sir. Actually, sir, for this is an in vitro study, so we used pure form of the cinnamon oil extract. But we are trying to develop a holistic, like a combined strategy using the biocontrol agent as well as the essential oil, like creating a formulation or something like that, so that the farmer can easily get it available. So the work is continuing, and we have even checked for the effect of the biocontrol agents like trichoderma and all with these cinnamon oil, and there is prominent results are there. So we are yet into that only. That's why I haven't included that slide here.
Okay, any question from anybody?
I have one question. You see, in conclusion,
yeah,
there is second person can serve as effective biocontrol strategy against Lactodiplodia theobromine.
Yes, sir.
Will you prefer to call this effective biocontrol or effective botanical?
Actually, botanical only, sir, but it can also come under biocontrol strategy, sir. That's why I have termed it as a holistic term. But it is actually botanical only.
Okay, thank you.
Thank you, sir.
Why you have given the questionnaire? Can.
Yes, now 7 is present. Serial number 7.
So, sir,
Yes, sir.
Can I stop presenting?
Yes, yeah, please you stop your
Okay.
screen
Okay.
share.
Thank you, sir. Thank you so much.
Now 7 is
present.
Jasmita.
Yes, sir, I'm here. Yes, sir, I'm here, sir.
Okay, you share your slide.
Sir, is my screen is visible, sir?
Small. You increase size.
Okay, sir.
Your background color is
Shall
very
I
light.
start, sir?
Background color is very light.
Sir, shall I start, sir?
Yes, start.
You made in landscape, but
Good
your mobile is in portrait.
Sir?
Yes, start now.
Good afternoon to all respected dignities and participants. Myself, Jashmita from Dr. YSR Horticulture University. Today, I'm here to give presentation on utilization of natural products for the management of post-harvest decay in horticulture produce. Fresh fruits and vegetables have low pH, high moisture, and nutrient composition, which make them highly susceptible to pathogenic attack. As a result, considerable post-harvest losses of fruits and vegetables are brought about by decay-causing pathogens. With the availability of limited land resources for agriculture expansion and the looming food security threat posed by the global climate change, reduction in post-harvest losses would be the option to meet the present and future needs of the ever-increasing hungry and undernourished population. The use of synthetic fungicides has faced limitations and restrictions owing to their tetragenicity, acute residual toxicity, and other effects on the human health and environment. Biodegradable and eco-friendly natural compounds such as flavor compounds, acetic acid, jasmonides, glucosinolates, essential oils, and active principles of some plants have gained importance and attention for their use as decay control agents and safer alternative to synthetic chemicals. Some of the natural compounds are acetaldehyde, acetic acid, jasmines, essential oils, and textiles.
acetaldehyde. It is a flavor compound produced by the ripening fruits. Such compounds have been used for post-harvest decay control. And acetic acid is a metabolic intermediate that occurs naturally in many fruits and used as an effective fumigant for surface sterilizing a wide range of horticultural produce. It is very effective in preventing post-harvest fruit decay caused by the post-harvest pathogens like Penicillium digitatum, Botrytis cinerea, Penicillium expansum, and Rhizopus tolerans, and jasminids. These are naturally occurring plant growth regulators, belongs to the class olipines. They play an important role in activating signal molecules in plant defense responses against the pathogen attack and activate the genes encoding the antifungal proteins such as thionin. Methyl jasmonate is a major derivative of the plant hormone jasmonic acid, and it plays a critical role in inducing the resistance to fungal pathogens. Essential oils that are derived from various plants have anti-pathogenic activity and controls the phytopathogenic microorganisms. For example, essential oil extracted from the lemongrass play a critical role in controlling the post-harvest pathogens like Botrytis cinerea and Aspergillus niger. Plant extracts. These are the bioactive compounds extracted from the plants, and they have the great potential as novel fungicide source for controlling the pathogenic fungi. Coming to the conclusion, over the years, a wide range of chemicals have been used to control the post-harvest losses in horticulture produces such as fruits and vegetables, and the ill effects of these chemicals on human health and environment have prompted the search for the safer alternatives. Post-harvest control. Natural products have been actively examined for their antimicrobial activity. Encouraging the result in this regard have indicated the potential for the development of natural fungicides that could be as an effective synthetic chemicals for post-harvest decay control. But among all these compounds, natural compounds, jasmonids are showing the good effective against controlling the post-harvest decay. Thank you, sir. Thank you for your kind attention, sir.
Okay, thank you very much for your presentation. But in your poster, you did not show any figure, any photo. That's why it is not looking good. Anyway, you have tried your level best, but that is the review paper. No? There is no your any practical research. You are the PhD scholar which year? Second year or first year?
First year. Presentations.
What will be your PhD program?
In tomato and brinjal grafting work, we have done. Means I'm going to do grafting work in tomato and brinjal, sir. Again, is soil-borne problems. Till now, I didn't start it, sir.
And both the speaker, both the author and the PhD scholar. Who is your chairman? No name of in this paper.
Because it is not my original research paper, right, sir? That's why I didn't mention.
The teacher should be your teacher or any faculty should have been included.
Okay, sir.
Yeah. Okay. Take care. Now next person, Dr. Tushar, who is
it is 24. 24 is present.
24. Okay.
Hasim Manzoor.
Yes, sir.
Yes. Share your screen.
Am I audible, sir?
You are audible, but where is your where is your slide?
Sir, my slide is visible?
No, it is not visible. You share it?
Sure.
Harinder Kaur Vidya, Legal Science and Karindra, Atharva,
Yes, it is coming.
Yes.
Okay, sir. May I start now, sir?
Yes.
So very good afternoon to everyone. Myself Hashim Manzoor from RIMD University, Mandi Gopinagarh, Punjab. So on the behalf of my major advisor, Dr. Gurvinder Kaur and Ms. Pooja, I am going to present my poster, which is about evaluation of selected fungicides and plant oils against Alternaria solani causing blight of tomato in Punjab. So here we are going for introduction here. The screening of 8 different selected oils against the test pathogen, that is Alternaria solani, was carried out to evaluate their antifungal activities. All these oils were evaluated at 4 concentrations, that is at 200, 500, 700 and 1000 parts per million for their antifungal activity against Alternaria solani using poisoned food technique. Each concentration of plant oil was replicated thrice and incubated at 22 plus minus 1 degree Celsius for a period of 8 days until the mycelial growth in control fully covered the medium in petri plates. The data on percent growth inhibition was recorded following Vincent formula given in 1947. After 8 days of incubation at 22 plus minus 1 degree Celsius in each treatment, the data were subjected to statistical analysis in completely randomized design, factorial using square root transformation as described by Gomez in 1983. Critical difference to determine the effectiveness of different treatment at 5% level of significance were also calculated by multiplying critical t-value with standard error. So when we are going to in vitro evaluation of fungicides, different concentrations of systemic fungicides, which is our avatar, combination of Zineb 68% wettable powder plus hexaconazole 4% wettable powder, Pavestin, carbendazim 50% wettable powder, and Rocco thiophanate methyl 70% wettable powder, and non-systemic fungicides, that is dithane M45, mancozeb 75% wettable powder, and anthracol propionib 70% wettable powder were screened against Alternaria solani using poisoned food technique given by FAC in 1907. These 500 fungicides were evaluated at concentrations of 50 parts per million, 100 parts per million, and 150 parts per million. And when we are going this observation is, we have seen that maximum mycelial growth in millimeters was found to in the fungicide that is Rocco and anthracol, and minimum mycelial growth was found in dithane and avatar, and vice versa mycelial growth inhibition was found in avatar and dithane, and minimum mycelial growth inhibition was found in Rocco and anthracol. Now in vitro evaluation of 8 oils like whose names are Cymbopogon nardus, Cymbopogon martini rosib, Origanum vulgariale, Rosmarinus officinalis L, Azadirachta indica L, Eucalyptus globulus L, Brassica junicea L, and Oleo europea L were evaluated against Alternaria solani by poisoned food technique, and data is presented, and from the data we can see here we have seen the maximum mycelial growth was found when we evaluated the oils, and it was found to be in Oleo europea, Brassica junicea, and Eucalyptus globulus, that is, and here their mean comes out to be 90, and the minimum mycelial growth where mean turns out to 0,
that was found in Symbopogon nereidus L, Symbopogon martina rosib, Origanum vulgare L, and vice versa. Maximum mycelial growth inhibition that was 90% found in the oil is Symbopogon nereidus L, Symbopogon martini rosib, Origanum vulgare L, and minimum mycelial growth inhibition that was found in the oil is like Oleo, Europea, Brassica junici, and Eucalyptus globules. Thank you. Thanks for your kind attention.
Doctor, what is the title of this presentation?
Sir, my title was Evaluation of Selected Fungicides and Plant Oils against Alternaria solani causing blight of tomato in Punjab.
When you have selected these fungicide and more than 50% of these are now, they are under ban. Carbendazim, Rocco, all these are, and Dithenam 45, all these are come under ban.
Sorry, sir, I am not getting what are you saying?
I am saying that these fungicide which you have evaluated,
Yes,
they
sir.
are now banned for use in India.
Yes, sir. Sir, I am right now pursuing masters. I have collected the information from previous research papers.
Oh, that is your master degree program. What?
Yes, sir, I am right now pursuing my masters.
Oh, you are doing master degree. That's the you have completed your writing thesis.
Yes, sir, I have not yet completed. Now trying to write.
Oh.
Sir, I have collected this
Your
information
MS?
from previous research papers. Actually, want to be part of this prestigious conference. So I tried to be part of this conference, and I searched previous research papers and got information from there.
Very good. Very good. Very good. That's why you could not give good explanation to Dr. Mishra. Okay. Now in the PhD program, you will do your best.
Okay, sir.
Thank you for your presentation.
Thank you, sir.
Now 25 is also present.
Next.
25 is present.
25.
Excuse me, sir. Sir, myself, Dr. Vinod Upadhyay. I am also.
Yes, no problem. It will be done because that time you are not there, so we moved there. We noted. You send the information. We noted.
Good afternoon, sir.
Gurvinder Kaur.
Yes, good afternoon, sir.
Yes, good afternoon.
Sir, is my presentation?
Yes, visible.
Yes.
Okay. Okay. Good afternoon to all of you. The today's topic of my poster presentation is evaluation of selected botanicals against the botrytis cinerea, causing a grey mold of glandulus in the Himachal Pradesh. In these picture, you can simply see that there is some symptoms showed by botrytis cinerea on the petals, on the different leaf parts of glandulus, like on florets, stems, leaves. And after some time, after surveyed the different field in the Himachal Pradesh, we simply just collected the isolate. Then isolate should be maintained under the laboratory condition, and their growth is observed on the potato dextrose agar medium. We can easily see that there is plenty of sclerotia made by botrytis cinerea on the PDA. After these pictures are shown that electron microscopy of botrytis isolates. In this, we simply just selected the different medicinal plants through which we can simply inhibit the growth of botrytis cinerea, like some different medicinal plants like Bana, Basuti, Karipata, Eucalyptus. These are the different medicinal plants which is easily available under the natural condition in local communities near about in Solan. And in this picture, you can easily see that the maximum mycelial growth inhibition was done by alien settlement, followed by the Acidiphyta indica, and the maximum mycelial minimum mycelial growth of Botrytis cinerea we can see on PDA, that is inoculated with the botanical extract of Allium sativum. Now, this picture depicts the different patterns of Botrytis cinerea on the different 8 medicinal plant extracts. And on the other hand, we can easily see on this tables that the minimum growth inhibition was shown by minimum growth inhibition was shown by the cow urine, butter spore, and the vermi wash compounds, and the maximum was by Allium and followed by the Azadirachta indica. Thank you so much, sir. Hello. Am I audible?
Yes, you are audible. Here in this petri plates, left side of your this electron microscopy.
Yes, sir.
In petri dish, the black color, what is that?
Cultures. You have shown it below material and methods.
Yes, sir. I can see on the left side, on the right side of mine, that is a scanning electron microscopy picks of Botrytis isolates.
Yes, I am.
We can.
I am asking about the left side of electron microscopic photo. Between this leaf symptom and this electron microscope, they are in petri plates. Petri plates, what is that? This is black color.
This is a sclerotia on the PDA medium, sir.
It is similar like Sclerotinia sclerotiarum.
Little bit, sir.
Okay.
I have actually surveyed the different fields and isolated near about 15 to 16 isolates of Botrytis cinerea. Almost all the isolate can easily form the sclerotia on the PDA except 2 or 3 isolates.
Yes.
And Dr. Jadhav, any question?
Okay, sir.
Okay, thank you, sir.
Thank you very much.
Thank you
for
so
your
much.
presentation. Nice presentation.
Thank you, sir.
So what is the full name of RIMT University?
Regional Institute of Management and Technology, sir.
Oh, yes. Good.
Thank you so much.
You are assistant professor there.
Yes, sir. I'm assistant professor here from last 6 years.
And Manjuri is your student.
Yes, sir. Hashim Mansoor is my student.
MSc student. Your first student or second student?
Sir, my third student.
Third student. Okay.
Yes, sir.
Thank you.
Thank you so much, sir.
Thanks.
Now 18 is present.
Now please stop your screen. Now
18. Serial number 18.
Vinod Upadhyay.
Yes, sir. Good afternoon, sir.
Share your screen, Vinod.
Sir, I have shared, sir.
Oh, okay. Okay.
A very good afternoon to the respective chairman, sir, co-chairman, sir. Today I am going to present my poster presentation on management of sun-scaled damage in pineapple by letting the shedding effect from naturally grown weeds. So, sir, pineapples are susceptible to heat stress and the sun damage, which can significantly reduce the marketable value of the crops. And sunburn is a seasonal phenomenon for pineapple and occurs above all during very sunny periods of summer, during the month of May to July. So from this, due to sunburn, it causes more hamper than the plant disease. Like it causes 30 to 50% damage every time if we make a clean crop or a weed pineapple crop. So that is why the present study we have conducted to see the shading effect of naturally grown weeds for the management of sun-scaled disease in pineapple. So here what we have done is that we have chosen the pineapple orchard which is in our station, And we have taken the pineapple plants that have even growth and even flowering were chosen for avoiding the errors of different sizes and maturity of fruit vigor. Pre-harvest following of treatments means here the treatments are followed, means one plot we have cleaned, complete cleaning of weeds, and in one plot we have allowed the 50% growth of the growth of weeds for shading up of fruit crop, and in one of the plot we have gone for no weeding at all, means 100% coverage of weeds after the flowering phase has ended. After the flowering phase, before that we used to do weeding, but after flowering we stopped weeding, and 100% weed shedding, 50% weed shedding, and no shedding. These are the three treatments we have followed, sir. And we have found that when we have gone for clean, there is no weeds, no shedding, then there is a 34.77% sunburn incidence. And we have taken it through the average number of 50 numbers of pineapple fruit. So 50% of shading of fruits by weeds, we have seen that 17.77% of sunburn incidence. But it is very surprising that when we go for full coverage of weeds after completion of the flowering phase, that is 100% shading, there is no sunburn incidence at all. So we have seen that the percent of reduce in sunburn incidence over the control, that is where there is no shedding, that control. So in 50% shading, we can reduce the incidence by 17%, and in 100% shading, we can reduce the sunburn incidence by 34.77%. And we have also observed, sir, that when we have taken the fruit weight as well as crown weight, we have seen that where there is a complete cleaning, no weeds are there, in that case the crown weight is high, which has no value, not that much value, and fruit weight is less as compared to 100% shading of fruit by weeds and 50% shading of fruit by weeds. So in this case, we want to suggest that, and even the similar, like sir, I want to support my result with the Safani et al. 2020 works. He also got the reported similar treatment when with 50% black shade net, there is no sunburn incidence as compared to treatment with fruit cap showing 5% sunburn incidence and 50 and when there is a no means clean, there is no coverage or nothing, then there is a 50% sunburn incidence respectively. Safani also got. So from the present experiment, it has been concluded, and I want to conclude it from the 2 years observation, that if you go for 100% shading naturally from naturally grown weed after the completion of flowering phase, then you can reduce the incidence of sun-scaled in pineapple by 100%. So you can get a very good marketable value, and there is no reduction in the marketable value, and there is no labor cost also. So these type of things should be, it is very easy for implementation in the farmer's field. And from these words, I want to conclude my experiment. Thank you so much.
Hello.
Hello, sir.
Vinod.
Yes, sir.
Dr. Jadov.
Hello.
What? Please. Any question?
ठीक है sir.
Because I am the virologist now, I don't have such kinds of knowledge that you have. That's why you should ask one question to him.
ठीक है sir.
ठीक है?
हाँ.
Okay. Thank you, Vinod, for your nice work.
Thank you so much, sir. Thank you. So much.
Okay. You are the PhD student?
No, sir. I am junior scientist from Assam Agriculture University.
Assam Agriculture University?
Yes, sir. Yes, sir.
Oh, Vinod, I have seen you. I forgot.
Yes, sir. Yes, sir. I'm
last year you came, na, for
yes,
this.
sir. I'm really happy for seeing you.
You know, but when you were speaking, we are not getting the photo. That's why.
Okay, sir. Okay.
Hmm.
Yes, sir.
Okay. Take care.
Yes, sir. Thank you, sir. Thank you so much.
Now, Dr. Tushar, who is the
Now,
next?
now in the list, I did not see anybody. If anybody is left, they can inform. If anybody is present for session 3A, any poster presenter is there?
Only 9.
Yes.
There's no any other.
Dr. Biswas,
Yes.
I can tell you the session 3B, they are present. If you don't want to waste time, you can start 3B also. Whoever there, you can take their presentation. Later,
Yeah.
if anybody comes, they will be entertained. Okay?
Okay. Anybody from session 3B?
Yes, there is name is there.
Akashdeep.
Hello.
Hello.
Sir, can I present, sir? Sir, my name is listed under 3B session.
3B. The serial number?
Yes. Just a minute, sir. Sir,
Your name
sir,
is?
my
6. 3B6.
yes, sir. 6, sir.
Akashdeep.
Yes, sir.
Okay. Start.
Okay, sir. Sir, just a minute, sir.
Share your screen, Akash.
Yes, yes, sir. Yes, sir. Just a minute. Sir, sir, is my screen is visible, sir, to you?
Yes, it is coming. Hmm. Okay.
So,
Yes, yes. Okay.
good afternoon to all of you, sir, respected sir, and all others. Sir, my name is Akashdeep Shutradhar. Sir, I am as MSc student of Lovely Professional University. So, sir, here today I am here to present my sir, research work of sir, on effect of selected botanicals on Trichoderma species in oyster mushroom cultivation. So, sir, this is my original research work of my MSc study, sir. So, in this study, I have worked on the disease of oyster mushroom that is caused by Trichoderma as a Trichoderma species, which is also known as the green mold disease of Trichoderma. So, sir, here I have observed all this in vitro and also under in vitro as a condition. I have, sir, I took 4 mainly botanicals for this purpose. That is a neem, wild sage, which is also known as lantana camera, then garlic, and sir, and then tulsi. So, under in vitro condition, I have prepared the botanical ethanol extracted botanical extract of all these botanicals for my study, and then I have applied it as per 5, 10, 15, and 20% concentration of each botanicals for my observation. And I have used for this, sir, I have used that for poison food technique for my this experiment. Under poison food technique, I have separately observed all the botanicals on the mycelial growth of Trichoderma, and separately as well as on the mycelial growth of Pleurotus ostratus. So, sir, after that, the same process was also used to evaluate this project on in vivo condition. So, sir, under in vivo condition, I have used the same botanicals, but this time I have used the dust or the powder concentration of all the botanicals as per the as I told, the concentration was 5, 10, 15, and 20. So, sir, in my presentation, you can say, you can see that, and these are the various results in which I have observed that lantana was one of the best result, which showed the best result against to control the mycelium growth of Trichoderma species. These are the various plates which you can see that in under 15 and 20% lantana percentage, it was able to control the mycelial growth of Trichoderma. And when I observed this on the mycelial growth of Pleurotus ostratus, you can also say that there was not much harm, that Trichoderma wasn't much harming the mycelial growth of Pleurotus ostratus. So by this we can conclude that lantana was the best treatment. And why I can say that lantana was the best treatment, in my all these graphs you can easily observe that this is my first graph in which it is showing that in vitro efficacy of selected plant extract on mycelial growth of Trichoderma species. So we can say that garlic and tulsi was not much effective to control over the disease, but along with that lantana was able to control inhibit the mycelial growth of Trichoderma. On the other hand, garlic was so much it was affecting so much on the mycelial growth of Pleurotus ostratus, but as the above pictures is showing, that lantana was very much familiar to the growth of Pleurotus under in vitro condition. And on the later graphs, you can easily say that I have evaluated for the time taken for complete mycelium run under in vivo, time taken for pinhead initiation under in vivo, and the selected botanicals, what was the yield. I also have noted down all these parameters for my study. And after evaluating all these, in the every aspect, lantana was proving the best treatment for all. It was taking less time took less time for complete mycelium run. It took less for the pinhead initiation, and it also was affecting best when I compare or when I took the yield, total yield, how much yield I got. Lantana was very much effective. So, sir, in my study, I actually my another object was to evaluate the cost benefit ratio by doing this. That how this experiment will going to help our farmers, sir. So here it was only a small experiment type. So here what I did, I took the total yield of all those percentage of bags of like 5%, 10%, neem, 15 and 20. And the total yield from all the neem treated bags was 1.15 kilo, and lantana 1.2 kilo, garlic was only 750 gram, and tulsi was on 700 gram. So as per a local market value, 1 kg of oyster mushroom is about it is selling price is about 200 per kilo. So in that case, after adding the prices of botanicals and all other expenses which we use to prepare the bags of mushroom, we can say that neem and lantana, as we was getting a good yield, it was also a profitable deal for the farmers and for as well as us. So, and as lantana and neem was actually affecting good to control about the disease in oyster mushroom, so we can say that lantana and neem are the best results on this aspect. So, sir, these are some of the pictures of our mushroom cultivation and all. This is a discussion part, and these are the every picture or data which I have included in my study and in this poster presentation. Thank you, sir.
Thank you for your nice presentation.
Thank you, sir.
Oyster mushroom.
Yes, sir. Yes,
oyster
sir.
mushroom and then Trichoderma species. How
Yes, sir.
related?
Sorry, sir.
Oyster mushroom and Trichoderma.
Yes, sir.
How it is related?
Sir, actually, Trichoderma caused one of the devastating disease in oyster mushroom, that is known as the green mold disease. That this green mold caused by Trichoderma species, it can eradicate almost the 100% growth of oyster mushroom. So it is one of the most devastating disease of oyster mushroom, sir,
Hmm.
in India today.
And how you will control?
Sir, for the control, sir, mainly I took, sir, all the botanicals like neem, lantana, garlic, and tulsi. So, sir, in my research work, I observed that lantana was best treatment to control over this trichoderma disease.
That's
And
the
it was
practical because you have shown
Yes, sir.
is effective. How will control? How will spray or what formulation? What kind
okay,
of
sir.
application?
No, sir. Sir, like in field condition, during preparation of the substrate, sir, during preparation of the wheat straw substrate or the paddy straw substrate, after the hot water treatment, we should mix all our botanicals in powder form. If it is possible in powder form, we should mix it with the substrate of wheat straw or with the paddy straw, and after the spawning, we can control. Like I mean to say thus, we should add all these botanicals while the bagging process.
Yes.
I have one question.
Anybody?
Yes, sir.
I have one question.
Yes, sir.
Waitress mushroom 200 rupees per kg. Where?
Sir, like actually, sir, I am sir, I have done my MSc from Sir Lovely Professional University, Punjab, and sir, I am belonging from Sir Agartala, Tripura. So, sir, it is sir. Actually, it is like market is very variable. It is not like, sir, we can say that, sir, market price is sir, ranging from 150 rupees to 200 rupees. The fresh oyster mushroom. So, what I have just taken average sir rate of that is 200 rupees per kilo of fresh oyster mushroom in local market, sir.
No, this
I
rate.
think no, it is not so costly.
Yes.
Huh?
Button mushroom at present, I think it is 200 rupees here in Delhi.
Yes, sir.
But I think West rice is not so costly.
Hmm.
Anyway, it is variable, it is sure. But you are doing research work in Lovely Professional University there, and you take the cost of Tripura.
No, sir. Actually, sir, the thing is that actually, sir, in Tripura market, sir, in Northeast, oyster mushroom are mainly preferable for the locals or for people
Yeah,
there.
that is true. That is true.
Yes, sir. So, and sir, in Punjab, there were no that much farmers. Actually, there were so much
Mostly they
little
grow
amount of farmers.
button mushroom. North India mostly button
Yes,
mushroom.
sir. Yes, sir. They prefer mostly button mushroom.
And another thing, this trichoderma that is growing directly on Pleurotus mushroom, or that is growing on the substrate of Pleurotus
Sir,
mushroom.
sir, it actually grow on the substrate of the Pleurotus ostratus. But the effect we can see it on the yield of mushroom. So if there is no yield, so we can say that this disease is devastating for the crop.
Yes, it is true. Actually,
Yes, sir.
trichoderma is completely covering the substrate, straw or wheat
Yes, sir.
straw or the rice straw. Because of that, the spawn run of Pleurotus mushroom or button mushroom, it cannot occur. Complete
Yes,
material
sir.
is covered by the trichoderma.
Yes, sir.
Okay, thank you.
Okay. Thank you, sir.
No.
Now 12 number is present. I saw
12.
others name I did not find out. If anybody is there, they can inform. I think 12 is present.
Uzma.
Yes, Uzma Faiz.
Hello, Uzma. Are you ready? Hello, Uzma.
His name is there in the participant list. He is present here.
Unmute. Next is
Serial number 12.
12. They know the timings.
Timing is given in both the cases, no? 3A, 3B, both the cases it is given, no? 14
Continue.
to 17 hours.
Hello.
Hello.
Hello. Sir, actually, serial number 7, actually, he is also from Sir Lovely Professional University, sir. He is just, sir, log in just in no time, sir. Just to wait for him.
Tushar.
Yes, sir.
Tushar is also there.
Yeah.
Yes, sir. He will join, sir. Like, I just call him,
Okay,
so he is just coming.
5 बजे तक तो ज्वाइन कर लेगा ना?
No anybody participant available. Perhaps.
Sri Kashmir University, Ejaz Ahmed.
You call one by one name.
Okay.
Yes, yes.
R. Karan. Oh. Nilima Kumari.
Dr.
No,
Kajal के
only 21 participants, na? Neetu Singh, Vikas Sharma, Akash
This
Devodia.
Ujma is showing.
Who?
Ujma at number 12 is.
Okay, Uzma, now. Ujma, are you ready?
Unmute yourself.
Then Kartik Nakum, Ejaz Ahmed, Sahid Ahmed, Kaunsat Javed, Uzma, Bara number, Ramesh.
Ujma has started screen share. Ujma has started.
Acha Uzma.
Now Usma is present. Okay.
That is the. She is on mute, I hope so.
She is unmute, but she has not on his video or video.
Perhaps another person.
Uzma is talking, but
Azma not present. What? Only slide shared.
Good afternoon, sir.
Oh, okay.
Yes.
So,
Uzma.
let's start.
Sir, yes, sir, I am here.
Slide show. You do slide show.
Sir, is screen visible?
Yes, visible because you do slide show.
Yes, slide
Okay,
show.
sir.
Okay. Visible,
Can I
but
start?
Yes.
Yeah, it
Start.
is visible.
Okay, sir. Good afternoon. Good afternoon. And quality, and thus inflicting huge economic losses. Here is the symptoms of this disease, and the pathogen. Here is the pathogen which I have isolated in my research work, and this is the mycelium of the pathogen, and these are the conidia. Now coming to the one of my objective, that was the management of the grape anthracnose by chemicals. First, I have evaluated 5 fungitoxicants under in vitro conditions. 5 fungitoxicants at 5 different concentrations have been evaluated against this pathogen at 5 different concentrations. And among 5 fungitoxicants, carbentism plus flucelazole.
Just I manage the marks.
It proves highly effective in inhibiting the radial mycelial growth, which shows 100% mycelial growth inhibition. And the least was shown by azoxystrobin plus difenoconazole.
Major comprehensive, minor comprehensive, and qualifying fiber.
Now, these were the results as I have on the piece of photographs. Now on after that, the four effective fungitoxicants which were evaluated under in vitro conditions were further evaluated under in vivo conditions, and these fungitoxicants were sprayed at the best concentrations which were evaluated under in vitro condition and were sprayed at 5 different stages: at bud burst stage, then fruit development 1, fruit development 2 stage, and at by spraying. And among these four fungitoxicants, again the carbendazim plus flucloxazole, it proves highly effective, and it which shows the inhibition of the disease incidence and the disease intensity of 87.90, and on leaves and on twigs it shows the control in of 55.71%, and in respect to the check, which having 69.1% disease on leaves and 70.90 on twigs and 38.40 on berry. These were the photographs which I have taken on my trial. This is the diseased field, and this is the control. This is the control field, and this is the field which I have sprayed. These are the trees, and this is again next treatment, Captan plus hexaconazole, and this is carbendazim plus mancozeb. Here you can see the effect of various fungitoxicants, combi products in control to the respect of, as compared to the treated plot. And so the conclusion was that the pathogen I have isolated from the disease sample was characterized as morphologically as well as molecular level as Phaseoloma amplinum, which is the major causal organism of grape anthracnose. And under in vitro condition as well as under field condition, carbendazim plus flucloxazole, it proves most effective against this Phaseoloma amplinum, followed by carbendazim plus mancozeb. Thank you, sir. I am highly thankful to the organizing committee members, and my major advisor and the members of my mycology and plant virology lab. Thank you, sir.
Thank you, Ujma. You are assistant professor there, or PhD student, or MSc student?
So I am PhD scholar there.
PhD scholar. The second year or third year?
So third year.
Oh, then you are going to write your thesis, or
Yes,
what
sir.
your plan?
So I am writing thesis.
Oh, and you did your master there.
Yes, sir.
Where you have done your master?
Sir, Skostke.
Huh?
Skostke.
No, in which university?
Sir, Skost, Sherry Kashmir University, Shalimar K, Kashmir.
Yes. Now, Dr. Tushar, you ask one question to her. Because I am the plant virologist now, that's why I am not asking questions. Where is Tushar?
That time I was not here. You show me the title?
Title is, you show the title.
In Jammu Kashmir, their grape cultivation is there.
Yes, sir.
Where the grapes is cultivated in Jammu and Kashmir?
Sir, in Kashmir, the major it's cultivated in Gandarbal area, in Kashmir.
It is there.
Sir, I have the trial in MSc work.
Oh, you have worked in your MSc work?
Yes, sir.
Oh, that is you have presented, that is your MSc work.
Yes, sir.
How this anthracnose causing crop losses in this area?
Yes, sir. So can you please repeat?
Crop, how it is, how it causes loss, and what
Sir, it causes loss because, sir, as I have shown here, because it affects the all the succulent tissues, leaves, tendrils, berries.
No. Crop loss, that is your symptom, nah? What is the percentage of crop loss? That means.
So crop loss, sir, it has shown 80 to 90% crop loss. Sir, in review, sir, I have seen in review, it has shown in 80 to 90% crop loss.
80 to 90% crop loss. So how farmers will be cultivating?
Yes, sir.
if the 90% crops are lost, so how farmer will be cultivating?
Sir, because farmers they apply sprays because it is the most prevalent
Crop
disease
loss
on wheat.
means a data should be there. I mean, what is the total crop loss caused by fungus internationally?
Internationally, it is
Fungal disease, how much crop is lost due to fungal disease?
so fungal disease. Sir, it is 45 to 50%.
45% crop loss. And what is the weed? How much percent lost by weed? How much percent lost by insect? How much percent?
Sir,
lost
the major crop losses are shown by fungal pathogens because they are more prevalent.
Okay, chalo, ठीक है.
Okay.
Next, anyone is there in
Thank you, sir.
3A or 3B? Anybody present here for presentation?
Naujma, now please stop your screen share.
Okay, sir.
Okay, thank
sir.
you.
Thank you, sir.
Now, I will ask Dr. Biswas and Dr. Virendra Kumar. If it is here, we could have.
No, somebody was, Dr. Tushar.
Yes.
Somebody has given information that that 9 number.
9.
Number 9.
3B9. Aija Ahmed Dar.
No, some Dr. Imesh Jadhav told me somebody is waiting, will be joining.
Seven. Tushar P. Bhaggasya.
7.
Sutradhar informed that he will be calling. Lovely professor.
Hello. Sir, like I don't think so that he will join, sir. Actually.
Okay.
Yeah.
So,
Sorry, sir.
Dr. Biswas and Dr. Virendra Kumar.
Yes, sir.
I shall ask you take a cup of tea, then again join. Leave some time here. Okay.
Okay, thank you.
If you are here, we could have served, but it is in virtual mode. For changing your mood, you take.
Only virtual mood tea also.
Yeah.
Given. So very thank you.
You see that 2A or 2B, when time is over, somebody came and then presentation was, that time you participate, isn't it?
Participant is very low.
Yeah, very low and irregular also. So
only 1 or 2 present in the technical session 3B.
3B, I think only 2.
Oh, 2, yes. And 3A.
3A, it is better
9.
9, I think. 9,
9, okay, 9. So very low.
You just give a little bit time. Let us see if anybody join or not.
Okay.
When you
So
combine 3A and 3B.
Report should come separately. Report should come separately.
But in case of 3A, only 8 in my list, 8 only.
No, 9 is there.
So serial number 2, 3, 10, 12, 16, 18, 24, 25. Another
is 7, you mark 7.
Oh, 7, okay. Malika, oh, 7, na?
Yeah.
7 present, okay, yeah. Take, okay.
Then
So
it is 9.
9, okay. And 3B.
Yeah.
6,
6 and
serial
12.
6 and 12, okay. Okay, 2. Okay, that's all.
Shiva. तो उतना टेंशन क्यों लेते हो भाई? ठीक है, टेंशन क्यों लेते हो? सब हो जाएगा। ये है? अभी तो हो गया। है।
Dr. Jadhav.
हाँ जी
Hello.
डॉक्टर साहब.
I think nobody will come.
Yes, sir.
I think nobody will come. We have to close the session. Okay.
जरूर.
Now, yes. Now you give the
Dr. Umesh Jadhav,
हाँ जी.
he does not want to close. He's nice man.
डॉक्टर साहब आप चेयरमैनशिप में ये सेशन पूरी रात चलना चाहिए.
अरे तुषार बाग is the man.
I am just helping you. Otherwise,
तुषार बाग मुझे 15 मिनट्स पहले फोन किया जे और हमारा काल से ये ठीक नहीं है ये नेटवर्क.
आज तो नेटवर्क तो आज
चला
तो
गया था. तुषार बाग का मेल हमारा वो जंक में चला गया. फिर ढूंढा, प्रिंट आउट लिया, तो हुआ. तभी क्लोज करो. जादव सर.
हाँ जी.
जादव सर क्लोज करना नहीं चाहते हैं, क्या किया जाएगा?
सर मैं आपका थैंक्स कर देता हूँ, फिर करते रहना.
ठीक है. थैंक यू वेरी मच एम एस जादव.
नहीं वो
आपको थैंक्स देखा उन्होंने.
एम एस जादव है मेरा दोस्त बोलता है. पीके बाग हमारा क्लासमेट है, आप जानते हो बचपन से. जादव सर.
यस सर.
पीके बाग 84 से है
सर. हमने जब ये
आप हो 97 से.
नहीं,
फर्स्ट टाइम 97 में तो हम लोग मिला ना.
हाँ 97 में. यस यू आर करेक्ट.
97 एंड में ना.
डिटेक्शन ऑफ वायरस एंड वायरस लाइक डिजीज ऑफ फ्रूट्स.
अलावत, डॉक्टर अलावत. डॉक्टर पूर्ण भी आए थे.
यस 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 यस.
अरे तुम्हारा डॉक्टर साहब का इस बार देहांत हो गया, जो पंजाबी हमारा सरदार जी जो सर बड़ा जो बाल था.
कौन सा सर? मेरे को तो नहीं पता लगा.
जो आए थे हमारा ट्रेनिंग में.
ट्रेनिंग में एक तो मैडम आई थी शायद.
ना पंजाब से.
मैं रिकवर नहीं कर पा रहा सर.
वही तो लास्ट ईयर.
नहीं, आप उस ट्रेनिंग की कह रहे हो या अभी आए थे? कौन सा कह रहे हो?
हाँ वही ट्रेनिंग, जो ट्रेनिंग में आप थे उस ट्रेनिंग का ही बोल रहा हूँ.
अच्छा वो तो हॉर्टिकल्चर के थे डॉक्टर. आप समझ गया मैं.
थीन
नहीं. हाँ मैं समझ गया. वो हॉर्टिकल्चर के हेड भी रहे हैं, हेड ऑफ द डिपार्टमेंट भी रहे हैं हॉर्टिकल्चर.
ना पैथोलॉजिस्ट पंजाब का.
डॉक्टर टीएस थिंड की तो नहीं कह रहे हो या बीएस थिंड की कह रहे हो?
अरे जो हमारा ब्रेन कैंसर हुआ ना.
नहीं, वहां पर दो थिंड हैं. एक डॉक्टर पीएस थिंड है बैक्टीरियोलॉजी के, एक डॉक्टर टीएस थिंड है जो आपने फंजीसाइड पे उस पे काम, अपने प्रेसिडेंट भी रहे सोसाइटी के. किन का बोल रहे हो?
ना जिन्होंने हम लोगों का साथ ट्रेनिंग में आया था 97 में.
सर मेरे को जहाँ तक याद है वो तो हॉर्टिकल्चर के बंदे थे शायद.
हॉर्टिकल्चर थे वो.
बायोटेक्नोलॉजी या हॉर्टिकल्चर के थे. हाँ इस तरह का. ठीक है, आप सही कह रहे हो. हाँ.
पैथोलॉजी ना.
नहीं, पैथोलॉजी में तो ये दो ही थिंड थे. एक तो बहुत सीनियर है वो बीएस थिंड, बूटा सिंह थिंड. एक ये डॉक्टर टीएस थिंड जो अपने प्रेसिडेंट वगैरह रहे. ये
वो थिंग नहीं है.
हाँ थिंड नहीं है, वही कारण है.
हाँ थिंग नहीं है.
वो बायोटेक्नोलॉजी में थे जो आप बता रहे हो. बायोटेक्नोलॉजी के थे.
पैथोलॉजी को है तो हेड ऑफ पिछला साल उनका देहांत हो गया ना.
चल मैं कल पूछ करके बताऊंगा.
ठीक है डॉक्टर साहब, आप बंद करो.
हाँ डॉक्टर साहब, मैं फॉर्मल वोट ऑफ थैंक्स तो कर दूं.
अभी तो कोई है ही नहीं. पार्टिसिपेंट तो 7 है. आकाशदीप है लगता है.
नहीं, हमने थैंक्स तो आपका करना. आपने कितना एग्जॉस्टिव ये सेशन चलाया, कितना.
जल्दी करो. अभी मार्क्स डालना है.
डॉक्टर साहब यही तो कह रहे हैं. तो respected Dr. K.K. Vishwas and Dr. Virendra Kumar, we are very highly thankful to you both for conducting this exhaustive session and your patience that you have given every chance to all the presenter to present his paper, and you have pointed out very good queries from the participant for their future plan also, and you encourage the young birds to become a good plant pathologist. So on behalf of society and on behalf of our two convener, I am highly thankful to you both for conducting this session on chemical and non-chemical method for disease management. Thank you very much, sir, once again. Okay, sir.
Thank you very much.
Yes, sir.
Now I'm leaving.
Yes, sir.
Dr. Biswas and Dr. Birendra Kumar.
Yes,
I
sir.
also thank you. Both of you spend your valuable time here sharing this session. Thank you. Thank you, sir.
Okay.
Then, Dr. Sahab, we have to leave or this session will be closed from the host side?
Session is closed.
Yes, sir. Okay, sir.
Okay.